Hello and welcome. Welcome to this wonderful virtual symposium to launch the Barts Centre for Squamous Cancer. You're most welcome. I'm Paul Coulthard, Dean of the Institute of Dentistry here at Barts Centre London School of Medicine and Dentistry at Queen Mary University of London. We have a number of speakers for you this afternoon, some from our own university and some from elsewhere in the UK and even further afield. So I'm delighted that you'll be hearing a little later from our keynote address speaker, Professor Jennifer Grandes, Professor of Head and Neck Cancer Surgical Research at the University of California, San Francisco. Thank you for making the time, Jennifer. We really appreciate it and look forward to your address. Thank you. I am Professor of Oral and Maxillofacial Surgery and I'm an oral surgeon. So I know more about oral cancer than I do other squamous cancers, such as those of the esophagus, skin, lung, and cervix. But you'll be hearing more about what's common across these cancers a little later. And a big, unique aim of our centre is to increase research into common pathways of genomic, transcriptomic, and clinical significance across these squamous cancers. You can see here the state of play regarding oral cancer. And you can see that the numbers are sadly increasing and that, unfortunately, awareness of this disease is not as common as it ought to be. We know that there's increased oral cancer numbers in areas of deprivation, and particularly so for men. We know something of the risk factors. We know that smoking, alcohol, diet, viruses and palm are involved, but we don't understand enough, and prevention, detection and treatment are poorly effective. You can see here the WHO figures from last year about oral cancer and its significance around the world. And in particular, the mortality picture on the right shows the high prevalence and unfortunate death rates in Asia. Our dental school is situated in the East End of London. It's a great dental school. We've risen through the ranks of late, so we're currently 11th in the world. And I have to say, I'm very proud that in our citations, we are first in the world. We also did very well in the UK government's research assessment framework last time round, and we hope to do well again. Our research is structured in these three ways, and you can see that we do already have oral cancer amongst these. We have a number of good researchers undertaking wonderful research, but our aim is to increase collaboration across our faculty, university, and beyond. Our mission for oral cancer research within our Institute of Dentistry is to answer the overarching research questions as to who, how, and why patients develop oral cancer in the East End of London by a comprehensive database, a biorepository, genomic and transcriptomic analysis, and clinical trials, and to, of course, extrapolate that for the global good. We have around our hospital and school a large Bangladeshi population with almost half the local population. Whilst born in England, we know that some of these risk factors will abound, and sadly and inevitably there's a high incidence of oral cancer. When I moved to London 2.5 years ago, this was one of the key reasons why I chose to do so. And I, like many who work here, that East End London population gives us a particular opportunity to do something really worthwhile. The opportunity to do something through our research to improve the health of those locally, but also globally. A little bit more about oral cancer. You can see that the treatment is largely surgical, sometimes followed by radiotherapy. Oropharyngeal cancers tend to present later because of their location and are more likely to be at an advanced stage, in which case they may be managed with more radiotherapy and chemoradiation. But generally, surgery is the mainstay. Of particular note, as you can see here, is the survival rate, which is unfortunately very poor and has not sadly increased over the decades. So we need to do something about it. Surgery for oral cancer has its own problems. There are huge, significant functional consequences around basic requirements of human beings to have fulfilled and happy lives. Eating, drinking, speaking, hearing, facial expression can all be affected. Depends, of course, on the nature of the surgery and also the extent of it, and of course, based on when the patient presented and how significant the disease was. But effects on appearance and identity alone can be really unfortunate consequences for these patients. So we need to increase the survival and the quality of life for these patients. Now, I don't mind sharing with you that just over a year ago, I suffered cancer of the prostate, underwent surgery very effectively, and I'm doing really very well. And I have to say, I was less daunted by that, probably because there's been more research in that area and the surgical sciences have advanced. So this is an interesting slide that shows that there's been a considerable mismatch between global funding and disease burden. Some cancers are funded at a higher level than their relative burden, such as prostate cancer and breast cancer, and some cancers are underfunded, for example, oral cancer and esophageal cancer. So this is where Barts Charity comes in, and we're delighted that they've generously awarded just over £2.6 million to create the Barts Centre for Squamous Cancer. So this will be dedicated to improving detection, treatment, and improving the quality of life for patients with squamous cancers through cross-institute calibration and bringing together research groups with diverse expertise from across our School of Medicine and Dentistry to tackle the problems. It will be dedicated to work with the population of East London and to tackle important questions for them. And as I said earlier, to extrapolate that to the great benefit of those suffering these diseases globally. So these are my colleagues I'd like to introduce you to who have been critical to us obtaining this funding. Irene Lee, Professor of Cellular Molecular Medicine, who has done most of the work on our application to Barts Charity. Catherine Harwood, Professor of Dermatology, and David Kelsall, Professor of Human Molecular Genetics. And more, of course. So you can see here in our structure that we have many other individuals who have been key to putting together our plans to tackle the squamous cancer diseases. And some of them you'll be hearing from in this symposium. So I'm also pleased to say that we're working in collaboration with the Oral Cancer Trust, which is a charity. And of course, we want to work significantly with patients, those who have had this disease, have the disease, or know people who have the disease. So our next steps are to increase our research capacity by recruitment of new additional top researchers. We already have a number of researchers across our faculty, as I've already described, but we wish to recruit 3 outstanding new researchers in the areas of molecular carcinogenesis, translational cancer research, and the epidemiology of oral cancer. We're also setting up 3 core facilities with staff. One of them is a tissue bank with clinical database, also a laboratory support for in vitro and in vivo models, and bioinformatics core support, so that we're well set to proceed. So thank you so much for listening to my introduction. I'm going to hand over to Adrian Biddle to chair our next session. Thank you so much.
Great, thank you, Paul. That was a fantastic introduction to the centre, and I'm looking forward to the 2 speakers we have in this first session of this inaugural symposium for the centre. What we'll do is we'll have both speakers speak one after the other, and at the end, after their talks, we'll have a bit of time for Q&A. So please do type your questions into the Q&A. You'll see there's Q&A box in Zoom. You can type your questions into the Q&A, and then after both two speakers have spoken, we'll put your questions to both speakers. So our first speaker today is Dr. Bartomeu Colom, who's a senior staff scientist at the Wellcome Sanger Institute. Bartomeu is interested in stem cells, I think, in somatic mutations, cell competition, and early tumorigenesis in the esophageal epithelium. And also, before moving to Cambridge, Bartomeu worked at Clonal. So we'd like to welcome him back today, and we're really looking forward to your talk, which is titled "Clonal Competition in the Normal Epithelium During Early Carcinogenesis." So Bartomeu, whenever you're ready, please do start.
Thank you for that kind introduction. I hope you can all see the slides.
Yep, we can see your slide.
All right. Let me minimize myself. Right. So today I'm going to talk about competition between mutant clones within the normal esophageal epithelium and how this may contribute to early carcinogenesis. And we are using mouse models to investigate this. So in recent years, there's been an explosion of papers showing that human normal epithelia is colonized by mutant clones that carry somatic mutations in what is thought to be cancer-associated genes. And that happened in many tissues. So when we look specifically at esophageal epithelia in humans a few years ago, we found in our group that the number of mutations increases with age in this tissue. And when we look at which mutations are selected in this tissue, we found that either if you look at mutations per centimeter square or the coverage of those mutations within the tissue, so the area occupied by these mutations, we can see that Notch1 is the main driver, mutant driver in the normal epithelia of the esophagus. We can find other genes that are also in the Notch pathway, like Notch2, 3, but we also find very critical genes associated with cancer, like p53 and so on. When we look at the prevalence of these mutations is when we start to see differences, whether we find them in the normal tissue or in cancer. So, for example, for p53, we can see that the prevalence of the mutation increases with age, so the percentage of the mutant epithelium increases with age in the normal tissue. But this is always lower than what we found in cancer. And this is a squamous esophageal cancer. On the contrary, if we look at Notch1, the probabilities to find mutations in Notch1 in this tissue are much higher in the normal areas than it is in cancer. So this is very interesting, and it raises the question as whether some of these mutations could be actually protecting against carcinogenesis. So we wanted to investigate this further, and specifically we wanted to look at how these mutant clones evolved in the normal tissue, how they compete between them for space and survival, and specifically we wanted to know whether the competition between these mutant clones in the normal epithelium has a role in influencing early tumorigenesis or carcinogenesis. So for that, we moved to the mouse esophagus. This is a really lovely model, and it's got very simple structure and dynamics. Just very briefly, we know that all the proliferation always occurs in the basal layer. So it's a simple cell layer of cells that will divide eventually, and then would eventually stratify upwards towards the suprabasal layer until they are shed into the lumen and eliminated from the tissue. And in terms of the dynamics of the tissue, So we know that once the cells divide, and all of the cells in this basal layer have that capacity to proliferate, and they will eventually. So we know that once one cell divides, it's got two options. So the cell fate can be either to divide again and stay in the basal layer, or it can differentiate upwards. And at this point, the cell will eventually disappear. And we know that there's a fine-tuned balance between the number of cells that go each of these steps. So to maintain homeostasis, you need overall the same number of cells that will divide or differentiate. And we know that there are some mutations, particular mutations that alter that fine balance between differentiation and proliferation. And once cells are more tilted, for example, towards division, then the cells that carry these mutations will expand in the tissue. So we wanted to study all this mutational landscape and how these mutations interact with each other and compete and so on. So we devised this model in which we give to the animals the ethyl nitrosamine, and this is or 10 for short. This is a nitrosamine, and it's a mutagen found in the tobacco smoke. And so in the protocol, what we did was to give the animals oral in the drinking water this den for 2 months, and then we stopped the treatment, and then we left the animals to rest for a year, which one we collected the tissues and then sequenced them. So the idea here is that we introduce these mutations, and then these mutations, so cells carrying them form clones by spanning tissue. So we would be able to study all these events. And when we do that, both by a number of mutations and coverage of those mutations in the epithelium, we find that, again, like we see in humans, Notch1 is the main driver in the mouse. I must say that this is all in normal areas, so there's no mention of tumors so far. We can see other genes also in the Notch pathway, like Notch2 or ADAM10, but we also find P53, GetCal3, ARID1A, very similar to what we found in the humans. And when we look at the main driver, which is Notch, we can see, and we look at the mutations that are present in each of the domains of the protein, we can see really mirror images between the mutagenized mouse model and human tissues. So what we have here is a really good model in which we can introduce those mutations, and that really mimics what we see in humans. So next we wanted to investigate how these clones compete between them. And for that we use lineage tracing. So for those of you that are not familiar with this technique, very briefly, this is we use well, it's a technique that allows you to follow the fate of those clones in vivo and their dynamics. And very briefly, so we have these genetically modified animals that carry a CRE recombinase that is inactive, and they also carry a stop codon in front of fluorophore, in this case is YFP. So when we activate that wheel by giving the animal some drugs, then we activate this CRE recombinase that would eliminate, remove this stop codon and allow the expression of the fluorophore. So what we would see is cells that express that are fluorescent, and this is inheritable. So what we will see is that all the progeny of this original cell is also fluorescent, so we can follow their growth on time in vivo. So this is a real example of this. This is a control animals of this YFP, so no treatment here. And we induce the animals. 24 hours later, we can see these single cells that have been induced. And this is a very inefficient process on purpose, because we want the cells to be very low, so the induction to be very low, so we can follow the growth of these clones without any merging. And then over time, we're collecting the tissues, and we see how these clones grow on time. But also we see how they disappear, because those clones that leave the basal layer will be eliminated. We call those floating clones, but they just float away. we know that when we introduce mutations, that equilibrium, that balance between differentiation and so differentiation or division of cells can be altered. So, and this is a very beautiful and extreme example, really, that when we introduce a mutation like a mammal one, which is so this is a transcription factor in the notch pathway. So when we introduce this mutation, what basically we're doing is to block the notch signaling. So in this case, in this animal, the cells that are fluorescent, and you can see here, also carry this mutation, but the rest are wild type. And when we do that, when we induce this mutation, what we see is that clones start growing massively, really, compared to the wild type control conditions, and they take over the whole tissue really after 1 year. And I'm just mentioning this mouse because we're going to go back to it later on. So we wanted to investigate how the clones evolve in our then-treated animals. And so for that, we took this YFP mice, we give them then or the vehicle control, and then after that, and we stop the treatment, we induce immediately the YFP clones, and then collected tissues at different time points after that, up to a year. And this is what happened in control conditions. So we can follow the fate and measure the size of these clones. By the way, this is each of these is the full esophagus of the mouse. So what we do is to collect the whole tissue. We cut it longitudinally, and then we just flatten it down, and we can take confocal images of the whole tissue, pretty much a single cell resolution. So we can count thousands of clones per tissue. So what we can see here is that in then-treated animals, we have clones that are growing much larger than in control conditions. But something, if you notice, is that we can see much less clones in the then-treated animals. And we can quantify that. So we have here quantification of the area of those clones. And again, we can see that they grow much larger than control conditions, but also we can see that they disappear faster than untreated animals. So what happens here is that there is a strong competition happening after we give them to these animals. And that makes sense because we've seen before that what we're doing is introducing particular mutations, and the mutations that alter the dynamics of these clones will be then expanding in the tissue. And they do that by eliminating the neighbors, which are less fit than themselves. And competition, so it's always relative to what you have next to you. And it's a process where the highly competitive clones will be eliminating those less competitive next to them. And this is a concept also that is worth remember for later. So far, we've just talked about the normal areas in these tissues. But we know that then is not only mutagen, it's also carcinogen. So we will be introducing and generating tumors in these tissues. So we know that most of the tissue will remain normal. We calculate that about 98% of the area of the esophagus will stay normal, as you can see in these images here. So tissue is flat, no structures appearing from the beginning to 18 months later. But we see sporadically these discrete structures that we can then analyze, and we define it by differences in the structure of the tissue, and they are staying positive for keratin 6, which is a marker of stress and is highly expressed in tumors. So what we have here is following then, most of the tissue will remain normal, like in these areas here, but some cells go rogue and start to proliferate or form these aberrant structures that we can detect from very early time points. These are very, very small. So this is about 50 microns, and the cells are composed, these tumors are composed maybe about 20, 30 cells at the very beginning. And the advantage that we have here is that we can look at the whole tissue at single cell resolution. So pretty much we can count all the tumors that are present from very early time points. And we can see how these tumors are growing. As you can see in these images over time, we can quantify the growth of these tumors, and crucially, we can count them. And here's where we start to see very interesting things, because we were very surprised when we found that at the very early time points, we detect a lot of these baby tumors, we like to call them. So recently originated tumors. So these are early dysplasias, and we count hundreds of them per tissue. And the interesting thing is that when we count them at later time points, we can see that their numbers start to decrease very quickly, actually, to the point that by 18 months, we only retain about 4% of these, the originally generated tumors. So we thought this is very interesting, because we want to know why this is happening, because we can harvest that mechanism, and maybe we can sort of prevent or act on it and make tumors disappear from the very, very early stage. So there are several ways how these tumors could be eliminated, and I'm not going to go through all of them, but I can quickly tell you that these early tumors are not being eliminated by apoptosis. We are unable to detect any apoptosis in these cells. They are not eliminated by a lack of cell proliferation, and definitely they are not eliminated by the immune system. And in fact, the immune cells are not able to detect these tumors at these early time points. So what is left? Well, I've just been talking about the normal areas in these tissues, and how the different clones are competing between them, and how the highly competitive clones can eliminate those that are less competitive. So we thought maybe that's what's happening, and these same clones in the normal areas that are highly competitive, very fit, could also be eliminating those early tumors. So this is what we hypothesize, and in this scenario, we can devise these three possibilities, depending on the fitness of the normal clones surrounding the tumors, and the fitness of those tumor cells. So in the first scenario, the tumor cells would be fitter than those cells surrounding it, and in that case, the tumor will grow. On the contrary, if the surrounding cells are more competitive than the tumor, then this tumor will eventually be eliminated and regress. And finally, if both cell types in the tumor and outside the tumor are equally fit, then they will be competing in equal terms, in neutral terms, and then just by chance, some of them will be eliminated, some of them will stay in the tissue. So we thought long and hard on how to demonstrate this, and we have a critical experiment, and we thought that it would be really, really good if we could manipulate the normal clones in the surrounding epithelia and making them more competitive, and see whether we end up having less tumors. And for that, we go back to the mammal mutant animal that I was mentioning earlier. So in this animal, what we thought the idea would be to introduce the mutations to generate those lesions here, and only after that, so 10 days after stopping the DEN treatment, which is where we have the lesions at the maximum point, if you remember, only then we activate this mutation and induce these clones. So the idea would be that once these clones start expanding, and we know that they are very competitive, they start expanding in the tissue, they are eliminating their neighbor clones, and when they encounter these early tumors, they will also be eliminating them. So what we would expect to see, if that was true, is that in the induced animals, we would detect less tumors. And this is exactly what we see when we collect those tumors some days after induction. But not only that, but also because the induction of these tumor cells, of these mammal cells is very variable. We have a range of induction level in these animals. So when we collect the tissues, we can measure the area that is covered by these mutant clones. And this is very variable. Each of these is one tissue, one animal, and the range that we have in the mammal area is about from 8% up to 35%. But this is very convenient actually, because we can then do some sort of correlations between the expansion of the mutant clones of the mammal areas and lesions that we found in the tissue. And when we do that, we can very nicely see that the more mammal we have, the more those clones expand, the more lesions are being eliminated. And this is proof that those highly competitive clones are actually really doing their job and eliminating those early tumors. So just to summarize that, the message take home really is that the mutant clones in the normal epithelium may have an anti-tumorigenic role in eliminating those early neoplasm by cell competition, and therefore they can preserve the tissue integrity. And conceptually, this is very interesting because it's telling us, it implies that the survival of these early tumors not only depends on the mutations that themselves carry, but also on the mutations, the mutational landscape of those cells that surrounding in the normal tissue. And we've shown that we can manipulate the clonal architecture of those normal epithelium. So this could have a role in the treatment on early cancer prevention. So that's all from me. Just thank you to all the people in the team that has contributed to this work, especially Phil, to the collaborators and the funding bodies. I'm more than happy to answer any questions after the next talk.
That's great. Thank you so much for really. Sorry, just a little bit of feedback there. Thank you so much for a really interesting talk there. And yes. Please, everyone, do type your questions into the Q&A, and we will then be able to put your questions to both Bartolomeo and Ines Quera at the end of the session. So we'll now move on to our next speaker, Dr. Ines Quera, who is a group leader in the Institute of Dentistry here at QMUL. Before joining QMUL in 2020, Ines spent 6 years working with Professor Fiona Watt at King's College London, where she established her own line of research using a mouse model of oral cancer. And here she determined the dynamics of tumour formation, and this is something she's taken on to her new group. So we're really looking forward to her talk, which is titled The Role of the Mutational Landscape and the Immune System in Modulating Oral Cancer. So Ines, whenever you're ready, please do take it away.
Hi, good afternoon. So first of all, I would like to acknowledge the fantastic support from Barts Charity to create a new center for squamous cancer that brings together the community of researchers working in different types of squamous cancers, from skin, head and neck, and esophagus. And I would also like to take this opportunity to thank the charity for their support to set up my lab that started last year at Queen Mary. Part of my team focus in oral cancer. And oral cancer is a subtype of head and neck cancer and includes the tumors in the oral cavity. It is considered the 6th leading cancer by incidence rate, and only 50% of the patients with the disease will survive more than 5 years. And this incidence is increasing, and despite the advances in treatment, the survival rate has not been improved in the last 40 years. While most head and neck squamous cell carcinoma are associated with HPV infections, the major risk for oral cancer are the use of alcohol, tobacco, and pen. And as Paul mentioned, it is commonly used by South Asian communities, and a particular problem in the East London community where our lab is based. Oral cancer is difficult to treat, and disfiguring surgery is often needed, and patients may lose their ability to speak, to swallow, to eat, and change their facial expression and appearance, and not being able to identify themselves in the mirror. So this leads not only to physical implications, but also to a very heavy psychological burden. So we need to better understand oral cancer, to find new tools for early diagnosis and to reduce treatment morbidity. Cancer is an heterogeneous disease that differs between patients and among different regions of the same tumor. And this intra-tumor heterogeneity is one of the biggest challenges for cancer therapy, with several intrinsic and extrinsic key players that have a role in tumor progression, metastasis, resistance to therapy, and cancer relapse. Several studies have uncovered the genomic heterogeneity of tumors, which has been fundamental to identify the specific genetic alterations that can be targeted for therapy, as well as used as biomarkers for early detections. So today I'm going to give you an overview of two of our recent studies. The first study dissects the genomic alterations in oral SCCs and how these affect the tumor microenvironment. And on the second study, we'll focus on one specific mutation, and we investigate how the loss of a keratin affects the immune system and modulates tumor progression. In recent years, large-scale genomic studies using next-generation sequencing technologies uncovered the genomic heterogeneity of tumors. And this heterogeneity has significant implications for the choice of molecular biomarkers and implications also when we're making clinical decisions. So the state of art for head and neck squamous cell carcinoma mutational landscape has been analyzed in a handful of papers now. And as expected, P53 is the most relevant mutation in this type of cancer, as in many others, followed by mutations in CDKN2A, Caspase8, FAT1, and Notch1. The approaches currently adopted in human have primarily focused in the genetic lesions in frank tumors, rather than the mutations that occur during tumor progression. But if we want to improve oral SCC's early detections and predict the pre-malignant alterations in early stages lesions, we need to determine which are the mutations that appear in early stages and predict which of these mutations can actually become, which of these lesions can actually become malignant and develop into more aggressive subclones. So in order to study the early genomic changes in oral SCC development and to determine new biomarkers for early detection, when I was a postdoc with Professor Fiona Watt, I established a model of autologous oral cancer using 4-NQO, which is a synthetic carcinogen that mimics the effects of tobacco and alcohol injection. So mice received 4-NQO in the drinking water for 16 weeks, and we monitored the appearance and the progression of the lesions for a total of 28 weeks. Hyperplasia and leukoplasia or leukoplakia were visible from 6 weeks. And from 10 weeks, mice had raised white lesions corresponding to dysplasia. And from 16 weeks, full oral squamous cell carcinoma began to appear. Several groups used this model to test new drugs, but while it is known that 4-NQO mimics some of the morphological and molecular features of human oral SCCs, we have no idea whether it covers the genomic alterations that are actually observed in human. So we were able to use this model to see the development of the lesions and harvest samples every 2 weeks, covering the different grading of the tumors from hyperplasia to dysplasia and finally to invasive SCCs. So we use this model to conduct a multi-level analysis of oral cancer genetic changes, correlating these molecular changes with the tumor microenvironment heterogeneity. Since tumors can lose their epithelial markers, we didn't use any sorting strategy or any marker to select cells as seen in previous papers. So that could actually bias our analysis. Instead, we microdissected the tongue tumor sections. We microdissected from the tongue tumor sections the regions of the tumor that was sent for sequencing, and then adjacent sections were stained and imaged and quantified for several immune markers to analyze the heterogeneity in the spatial organization of these markers within the tumor microenvironment. The genomic DNA extraction and sequencing was done in collaboration with David Adams at the Sanger Institute. Bioinformatics analysis was done in collaboration with Alessandra Vigilante at King's College, and tumor subclonal analysis was done in collaboration with Trevor Graham and Mark Williams, the Barts Cancer Institute here at QMUL. So first, we analyzed the mutational spectrum of foreign NQO carcinogenesis. And overall, we have seen C2T mutations that dominated the spectra. T2C and C2A mutations were also common. We also analyzed the similarity between the mutational profile of each of the lesions here with the cosmic signatures. And I'm just showing you here the lesions that are moderate, severe dysplasias and invasive SCCs. And we have seen that they actually show an enrichment of the signatures related with human neural SCCs. So in particular, these tumors presented a high similarity with signatures 4, 18, and 29. And these are observed in head and neck, human head and neck SCCs, esophageal, stomach, gingival oral SCCs, and are also known to be associated with tobacco use. We next analyzed all the genes that were mutated in our samples and correlated the mutated genes with the corresponding biological information, including tumor grade, degree of differentiation and invasiveness, proliferation index, extent of immune infiltrate, and we focused on genes that are known to harbor these bona fide driver mutations in human tongue squamous cell carcinomas. So from the mouse tongue recurrent mutated genes, the highest ranked genes are P53, FAT1, SINE2, and NOTCH1. FAT1 was mutated in 30% of the tumors, particularly in moderate to invasive SCCs. And it is known that loss of FAT1 is associated with tumor progression and survival and gives competitive advantages to oral SCC cells in culture. P53 is a well-known tumor suppressor and was mutated in 30% of our samples. And these mutations were found in this place also in hyperplastic lesions in mice that were treated for long periods with foreign QOs, supporting the observation that loss of P53 alone is not responsible for tumor initiation. So with this analysis, we demonstrated that the mutational spectrum of the human oral cancer is recapitulated with this model. By comparing the mutational landscape with tumor ecology, we uncovered the correlation between NOTCH1 loss of function mutations and an increased number of immune cells in the tumor microenvironment. While mutations in the extracellular matrix protein laminin alpha-3 correlated with tumor invasiveness. Another big challenge in cancer therapy is the intratumor heterogeneity, and where different subclones of the tumor can actually carry different mutations. So a tumor may comprise subpopulations of cells, resulting from the expansion of a single clone with a distinct set of mutations. And these clonal subpopulations can coexist within a tumor for long periods of time. And usually when a tumor relapse is often from a subclone that actually survived therapy. In collaboration with Trevor Graham, we used a computational method to measure clonal dynamics and determine the subclonal architecture of the tumor samples. So what we found using the bulk DNA sequencing of our samples was that high genetic heterogeneity in invasive SCCs, we found that there is a higher genetic heterogeneity in invasive SCCs, where we see the emergence of a clone with a group of cells that will share the same subset of mutations. And this can also is a feature that can also be detectable in early stage lesions such as mild dysplasia. So most of our mild dysplasia lesions would have only neutral mutations. Here in this peak below variant allele frequency below 0.25. However, we did see some cases where mild dysplasias will see an emergence of a subclone, where there will be a subset of cells with a variant allele frequency around 0.5, similar to what was observed in the invasive SCCs. So this may be a feature that can actually be used to detect early lesions, and it will be an extremely powerful tool to detect this, for early detection of malignancy in mild dysplasia. Then, to broadly assess which mutated gene were represented in each clone, we annotated the mutated genes in each VAF, VAF peak. And we observed that in high-grade tumors, for example, bearing a clone, Notch1, FAT1, and SINE2 mutations had a similar VAF. So this will give us an information about the genetic heterogeneity within this subclone. So to summarize this first project, we performed an integrated genetic and phenotypic analysis of a mouse model for oral SCCs, demonstrating that it actually faithfully recapitulates the aspects of the human oral SCCs. We were able to determine this that they faithfully recapitulate the mutational signature seen in human oral SCCs. We were able to, since we were using a model and collecting tumors at different time points during tumor development, we were able to determine the genes involved in key time points in tumor formations and correlate these genetic changes with the biological features, as well as analyze their subclonal heterogeneity. So this can be an excellent tool for early diagnosis of premalignant lesions. I also focused in one particular protein that is known to be downregulated in oral SCCs. Keratin 76 is detected in normal epithelia biopsies expressed in the suprabasal layers of the oral epithelium. And in patients, as tumor progresses, there is a loss of Keratin 76. However, Keratin 76 knockout mice do not develop spontaneous oral SCCs during their lifespan. And it has been previously reported that loss of Keratin 76 in skin epithelium leads to hyperproliferation of the skin epithelium, an increase of the suprabasal layers, but we haven't observed any histological abnormalities in the tongue epithelia and no changes in the proliferation or tissue architecture. So altogether, this suggests that the genetic loss of Keratin 76 is not the sole molecular event leading into cancer here. So using the same carcinogenesis model with foreign QO delivered in the drinking water, we compared the incidence of oral cavity tumors in Keratin 76, in control mice, Keratin 76 heterozygous mice, and full knockout mice. And there was no significant difference between the control and the heterozygous Keratin 76 mice. However, Keratin 76 double mutant, the knockout mice developed oral SCCs earlier than the controls. So Keratin 76 knockout mice had an average onset of lesions around 12 weeks compared to 70 weeks in the control mice. And consistent with the findings in human patients, we did observe a downregulation of Keratin 76 expression in the tumors, in the lesions from hyperplasia stages. So Keratin 76 mice are more susceptible to carcinogen and develop tumors quicker than controls. The reason for the increased tumor incidence is the fact that keratin 76 knockout mice exhibit exacerbated local and inflammatory response to carcinogen treatment. There was significantly more stromal CD45 positive cells in keratin 76 knockout, together with an increase of the regulatory T cells and the decrease of the effector T cells in the treated regions. Effector T cells have an important anti-tumor role, while regulatory T cells are important in regulating T effector proliferation. Our hypothesis is that keratin 76, the increase of Tregs is actually dampening the anti-tumor response of the effector T cells. Immunofluorescence labeling for FOXP3 in the tongue tumors revealed an increase of the Tregs, both in the tongue keratin 76 knockout mice, both in hyperplasia and dysplastic tissue, suggesting that there was a higher inhibition of these effector T cells in the tumor microenvironment. So to discover whether the functionality of the Tregs and T effectors differed between keratin 76 knockout and control mice, we performed in vitro suppression assays. For those that are not familiar with this technique, what happens is that Tregs are isolated from the spleen of control and knockout mice and co-culture at different ratios with a fluorescent labeled CD4 positive, CD25 negative responder T cells, which include the T effector population. And what we observed is that the keratin 76 knockout Tregs inhibited the T responder proliferation more effectively than the control Tregs. And Tregs from the knockout mice also expressed higher levels of Treg suppressive markers, such as CD39 and CD73 when compared to the control. So overall, the Tregs from our keratin 76 knockout have an enhanced suppressive capacity. So to summarize what you know so far, the genetic ablation, this keratin 76 knockout mice exhibit this inflammatory phenotype with some systemic components. And we observed that there is a significant expansion of the Tregs. The Tregs have an enhanced suppressive capacity, and this leads to a higher incidence of oral SCCs. So this study demonstrates that keratin 76 is not just a cytoskeleton protein acting as a scaffold for cells, but actually is regulating the immunity in mice, and demonstrates its importance in tumor progression. And this supports the now well-accepted role for keratins as regulator of inflammation and immunity in skin, and shows a previously unidentified immunomodulatory role for keratin 76 and their importance in tumor progression. This project, this work on the immunoregulatory role of keratin 76 is currently being investigated by two very talented PhD students, Ines Tomas, that has worked with me at King's and continues under the supervision of Fiona Watt, and Mara Gelmetti, currently in my lab at QMUL. And Raju Kumar, a newly recruited postdoc in my lab with experience in cancer genomics and in vitro models, is focusing in dissecting human oral SCC heterogeneity. I would like to thank Professor Fiona Watt for her support while exploring these two projects while a postdoc in her lab, as well as the members that helped me during the work, in particularly the students that worked with me on these projects, Ines, Dido, Marina, and Ronan. This multidisciplinary work wouldn't have been possible without the help of several people to whom I'm very grateful. Alessandra, David, and Trevor and Mark I mentioned during my presentation, but also the inputs with their immunology expertise from Joanna Neves, Giovanna Lombardi, Paolo Bonfanti, and Dan Pennington that recently joined the project to co-supervise Mara on dissecting the role of keratin 76 in immunoregulator. And of course, the pathologists, orophathologists that bring their clinical view into our projects. And we are currently working with Anacotum that, and Raju is currently collaborating with Anacotum. And finally, I'd like to acknowledge the funding from the Barts Charity and Royal Society to my lab, as well as the funders for the fellowships for my lab members. And I'll be very glad to take your questions. Thank you for your attention.
Thank you, Ines, for a fantastic talk. That's brilliant. So what we'll do is we will, I've got some questions in the Q&A for Bartomeu, and we will go through those questions first. And then whilst we're doing that, please do write your questions for Ines into the Q&A, or if you prefer, you can raise your hand and we can invite you to ask a question verbally. So do feel free to do that as well. So the first question, Bartomeu, is a question from Ken, who asks, where does the clonal competition between mutations take place? Is it in the basal layer, or is there competition in both the basal and the suprabasal layers? And you're on, yeah, that's it.
Yeah, that's a good question. So competition, it occurs for the characteristics of the tissue, it occurs in the basal layer, because anything that goes to the suprabasal layer will eventually be eliminated for the dynamics of the tissue. So basically, it does happen at a level of basal cells.
That's great, thank you. I just want to maybe extend that by asking, do you find any of the mutations, you mentioned it, the MAM1 mutation, any mutations that can induce the cells to go back to the basal layer from the suprabasal layer, or are all your clones always started in the basal layer, no matter what the background?
Good question. So when we induce the mutations, either with them or by different mutant animals that we can induce, so individual mutations, I suppose that could happen both at basal layer and suprabasal. But as I said, the ones at the suprabasal layer will eventually be eliminated. So the result of those clones that we see at the end and over time will all be derived from the basal. To my knowledge, we haven't ever seen, and I don't know of, cells going back to the basal layer.
That's great, thank you. We have a question here from John Wang, who says, fascinating talk. Where does field cancerization fit in here? Does that not contradict to the process that you're talking about? So is field cancerization contradictory to this process?
Well, I don't know. It depends on what you mean by field cancerization. So what we're doing is simulating a lot the tissue, and it's undeniable that tumors, both in our model and also in human, it comes from mutations in those cells. But as we see, a lot of the tissue remains absolutely normal. So no phenotype there. So it's a tricky question to answer, really. Do we see field cancerization? Yes, in the sense that we have a lot of mutations, but no, in the sense that not all of these mutations will give cancer, and the tissue stays absolutely normal for 18 months after the DEN treatment. So yeah, I don't know if I answered that question.
That's okay. And John Wang, please do feel free to follow up via the Q&A or the chat with Bartomeu. We are quite short on time, so I'll ask one question to Eunice before we finish. But of the other questions in the Q&A, speakers, please do feel free to answer the questions by typing answers into the Q&A. You can do that. Just so after this session, but Eunice, just one quick question for you. I was very interested in this role of keratin 76 in modulating the immune system. Do you have any idea about what happens downstream of keratin? Presumably this keratin is an intracellular molecule. How does it influence the immune system?
We are working into hypotheses at the moment. We can actually follow the lead of Pierre Colombe, where there's more and more evidence that keratins can go nuclear and affect transcription of tissue self-antigens, for example, in the nuclei. And this has been seen, for example, with the mutation, loss of expression, well, mutants for keratin 17 that they show that keratin 17 protein goes into the nuclei in skin epithelial cells, and modulates the expression of AIR, which is a transcription factor important for the expression of tissue self-antigens. And so this can be occurring also with keratin 7.6. Another thing that can be happening, and this is something, one of the other hypotheses we're trying to dissect, is that we know that in skin, the loss of keratin 7.6 leads to an alteration of the epithelial barrier of the skin. We haven't seen that in tongue or stomach or even esophagus, and by playing a role with the tight junctions, so regulating the Claudin-1 expression. And so this can be happening actually in the thymus, where it's important to keep a normal architecture of the thymus. And keratin 7.6 is expressed in the thymic epithelial cells. So if there is an impairment of the migration of the T cells during T cell maturation, and there is an alteration of the negative selection of these T cells and the migration of these T cells into the peripheral organs, then keratin 7.6 can actually be impacting the different T cells at the thymus level, so in the maturation of the T cells. So this is still work in progress.
Great, thank you, Ines, that answer. And I see we've got some more questions come through for both Tomio and Ines. Unfortunately, we have run out of time in this Q&A, so if you both want to answer the questions by typing, that'd be fantastic. And I will now thank both the speakers. It's been a fantastic session. And now hand over to Professor David Kelsall, who will give us a little bit about Barts Centre for Squamous Cancer. David, are you there?
I'm here.
Great, excellent.
I was just fiddling around trying to get my screen shared, and I think I've done it.
Okay, you can see your screen. That's brilliant. Thank you.
So yeah, well, thanks for a great start to the symposium. And I also would like to thank Adrian for organising the whole symposium. He's done a great job so far. Yeah, so I'm here to set the scene for the ambition of the Centre for Squamous Cancer at Barts, and also to introduce our first keynote speaker. So, the Barts Centre for Squamous Cancer came about with generous funding from the Barts charity, and the work will take place between the dental school, which you can see on the right-hand side, and the slightly more colourful building, the Blizzard Institute on the left-hand side. We're both based at the Whitechapel campus in East London. So the ambition of this and aims of this new centre is to invest research and talented researchers into studying a relatively understudied cancer, the squamous cell carcinomas. They're the most frequent human solid tumours and are a major cause of mortality. And I think the focus at the centre at the moment, by looking at the individuals who are there, is based largely around oral, esophageal and cutaneous SCC. So as Paul Kuthald alluded to earlier on, we're with this generous funding from the Barts charity, we're aiming to increase research capacity to study common pathways of genomic, transcriptomic and clinical significance across squamous cancers from different tissues. So yes, that means we are recruiting academic group leaders. So we have 3 senior positions available, and we also will be looking to recruit support staff at the postdoctoral level or research technician level to help facilitate the work within this new centre. And one of the aims is to apply the findings, research findings, to benefit patients, of course. And we have particularly patients in East London, where there will be a focus on oral cancers, because these cancer types are of a particular problem in East London, as they are very common in patients of South Asian origin. So why are we working on so many different SCC types? Well, there's this thing called the squamousness of SCC tumours, and this comes about from a number of studies. What I will talk about is the data that comes from DNA sequencing. So this comes from data generated from either whole exome sequencing or whole genome sequencing. And it's sequencing a large series of different SCC types, from the lung, esophagus, cervix, head and neck. And as Tomo mentioned earlier, that what you see is common genes being mutated. So, and common pathways. So P53 is the most commonly mutated type across all SCCs, and then Notch follows a close second, being particularly highly mutated in cutaneous SCC and a little bit less so in the other SCC types. And when you look at all the genes that mutated in SCCs, what you see is this squamousness gene signature across the SCCs, where you see particular genes commonly mutated, but also you see genes that are not mutated at all. And so this suggests there may be some common mechanisms and disease pathways occurring between SCCs, regardless of the tissue of the origin. The other things that are emerging are there may be a common driving pathway leaders here. So in this case, I think SOX2, the transcription factor SOX2, seems to be driving a common lineage as well. So the idea of bringing people together from different areas and working different cancer types is we might be able to understand this squamousness of cancers. And already it's emerging, which you'll hear from the, I think Jennifer Grandis, the keynote speaker, will talk about as well, is that this is leading to sort of identifying potential therapeutic targets, such as the PI3 kinase pathways and mTOR, which may be not just a useful treatment strategy in head and neck tumors, for example, but may have utility in the other SCC types. So back to the Barts Centre. So as I say, we're building this critical mass in squamous carcinogenesis, and we have support staff and which will allow us to coordinate tissue specimen collection, clinical data, and also study epidemiological data, which will give greater insight into squamous cancer. And there'll be lab-based work going on, as we've heard already from Ines, and we'll hear later from Adrian, about exploiting our outstanding expertise in molecular biology and in vivo and in vitro modelling. So some may ask, why am I here? Because if you look at my CV, you don't see many papers on SCCs, and you're right. I mean, I did do my PhD in breast cancer, but then with the support of Irene Lee, I then focused largely on studying rare genetic disorders of the skin. But then this eventually brought me into SCC biology, because one of the conditions is called tylosis with esophageal cancer. And it's a dominant inherited syndrome where you get palmoplantar keratoderma, which is thickened hyperkeratotic lesions on the palms and the soles. But these individuals also have a very high risk of developing squamous cancer of the esophagus. And if they're not monitored, they could have their lifetime risk could be almost 90%. A few years ago, we eventually identified cause of this condition, the underlying genetic cause of this condition, and it was due to heterozygous gain-of-function mutations in a gene called RHBDF2, which encodes IROM2. At the time, we didn't know much about IROM2, apart from that it was an inactive member of the rhomboid protease family. However, we are now learning a lot more about the biology of this protein. So it's a 7-transmembrane protein. As I say, it was a protease, but through evolution, it's lost its protease activity. But it has this very long amino terminal loop, which is in the cytoplasm. And this is where the mutations that cause this inherited condition all cluster. So we've focused a lot of our biology in trying to understand about this unique amino terminus of this inactive rhomboid protease. And what we're finding is that it underlies, can regulate a wide variety of cellular processes. It can regulate inflammatory cytokine secretion and it's shredding via ADAM17. It can also regulate EGFR signaling, which is obviously a common pathway in many SCC types. It can regulate the barrier. So Ines was talking about the barrier there in terms of immune infiltration as well, so probably a link there. It can also regulate the cytoskeleton dynamics because it binds and interacts directly with type 1 keratins. And we also think it's got some sort of transcriptional regulatory activity, either directly or indirectly. It can regulate oxidative stress pathways, apoptosis, and we think we have new data suggesting it can actually regulate transcription factor biology as well. So it's just some data. So what we've So we have patients who have these inherited mutations in IROM2, and we have access to biopsies from these patients as well. So on the left-hand side, I'm just showing you some data taken from esophageal tissue biopsies. So these aren't cancers, these are just normal esophageal biopsies taken from the top patients when they go for their annual endoscopy. And what you can see from RNA-seq analysis is they have high levels of Notch1 expression. Equally, when you take skin biopsy, so this is uninvolved skin, it's not taken from the palm of the sole, and you can see they have constitutively high levels of Notch1 expression. So IROM2 in some ways is regulating one of the key squamousness genes of SCCs, Notch1. So going back to the centre, so one of the things we were very interested in, and Ines has already mentioned this, is we're trying to understand the early stages of human SCC development. From our work on IROM2 and looking at these normal, slightly hyperplastic lesions from the top patients, we've identified what we think is an early stage SCC gene risk panel. And when you look at the genes that are regulated by IROM2 in the esophagus across esophageal cancer data sets and also oral cancer data sets, you can see these genes are definitely regulated, definitely being deregulated in these tumors. So what we're trying to do is to nurture young talent. So we've already heard Ines speak, gave a wonderful talk, and Adrian will talk later on today. We already have some talented people in our centre and who are also bringing in external funding, which is also an important way to maintain the sustainability of the centre. So we've already heard about using genetic mouse models, but we're also investing heavily in technology and approaches to produce better in vitro human cell models. I think Adrian might talk about some of the microfluidics and organoids that can be developed. And it's a shame you're having to be virtual, because the Blizzard Institute, where most of the lab-based research takes place, is actually quite a quirky little building, quite interesting to see. And it would have been nice to have a sort of a cocktail after the symposium in the darkness to see the spiky hedgehog all lit up. So hopefully next symposium you'll be able to come to see us. So now it gives me great pleasure to introduce our keynote speaker, particularly as it's so early in the morning in San Francisco. So we're delighted to have Professor Jennifer Grandis here. Jennifer holds the University of California, San Francisco Distinguished Professorship in Head and Neck Cancer, and is also Associate Vice Chancellor for Clinical and Translational Research at UCSF. Her research focuses on improving treatment strategies for head and neck squamous cell carcinoma, with a focus on identifying and targeting key signaling pathways and genomic alterations. We're absolutely delighted that Jennifer can be here. It's a shame it's via Zoom, but it's still wonderful that she's here to give the inaugural keynote lecture in our new Barts Centre for Squamous Cancer symposium. We'd like to please, as Adrian said, put questions on the chat so we can ask a few questions after Jennifer's presentation. But so over to you, Jennifer. Are you ready to give your talk on the topic of head and neck squamous cell carcinoma precision medicine?
Yes, I am. Thank you so much. I just need to now share my screen, right?
I need to stop sharing.
All right, we have to take turns. All right, here we go. Let me make sure I get into slide presentation mode. And how is that, everybody? All righty. So it is such a pleasure to be here. I only wish I was there in person, not only because of the time difference, but your building looks so cool. And during the pandemic, I must confess, I've become an absolute addict for British television shows. So I would just love to be in England and tour London and see all my favorite spots and characters. But it's really a pleasure to be here today. I want to start by emphasizing, from my point of view, the incredible importance of team science, We don't do any of these things by ourselves. Academic medicine often emphasizes the glory of the individual in terms of career advancement, but it's my great pleasure now to co-direct a research group with my basic science partner, Dan Johnson, and my head and neck surgical partner, Patrick Ha, and together we are able to host a nice diverse group of trainees, and this is February in San Francisco, so that's the best part in some ways about living there. It's always lovely weather. I need to disclose that I have an investigator-initiated trial with support from Insight Pharmaceuticals. I will not talk about that today. I do some scientific consulting for startups in the Bay Area, and I will speak a little bit about a drug that I've co-invented that targets the transcription factor STAT3. So I have some patents on that. So I don't need to belabor this point for this audience. Your center sounds fantastic. Jill Siegfried and I, who is a lung cancer biologist, have always maintained that squamous cell cancer of the lung and of the oral cavity are very similar disease. And I'm sure squamous cells everywhere are very similar disease. We get balkanized by the types of surgery we do in the patients that we see. Today, I'm going to focus my talk mostly on oral cavity and then oropharynx and hypopharynx and larynx cancers, squamous cell carcinomas of the head and neck. But one thing that's really important to remind all of us about, there are very few virally associated cancers. And nasopharyngeal cancer caused by Epstein-Barr virus is endemic in southern China. And moving to San Francisco from Pennsylvania, I realized that the really large Cantonese population made nasopharyngeal carcinoma caused by EBV relatively common. And I will tell you a story that I'm sure you're all familiar with about the emergence of human papillomavirus as an important cause of oropharyngeal cancer. And those of you who are clinicians realize when you look in the back of someone's nose or mouth, there's no line separating the nasopharynx from the oropharynx. And so I think it's really compelling biologically that we have these two juxtaposed anatomic regions that are ripe soil for viral induced carcinogenesis, something we really need to learn about. There are two types of squamous cell cancers that arise in the head and neck. The most common, the type that I saw a lot of when I was training, is associated with smoking. Now, I do love this picture, but I will point out that if you get to be 100 years old and you're still smoking, you're probably not going to die of an oral cancer. But the HPV-associated cancers of the head and neck have really emerged as an epidemic. And to underscore that point, you know, this is an older slide, but this line just keeps going up and up and up. The number of HPV-positive throat cancers in men passed the number of cervical cancers in women in the US a few years ago. And last year, according to the National Cancer Institute SEER data, throat cancers in men associated with HPV was the ~1 fastest increasing cancer in the United States. And so when we look at our specimens, probably 80 to 85% of oropharyngeal cancers are associated with human papillomavirus. I will just say one final thing about this, then delve into the biology. This is, from my point of view, an entirely preventable cancer. I hope that in the United Kingdom and in Europe, you do a much better job of vaccinating your boys and girls before they're exposed to HPV. It's quite chaotic in the United States, and the vaccination rates overall for girls are about 2/3, but for boys, it's still only about 1/3 of our boys are being vaccinated against HPV prior to exposure, which from my point of view as a cancer doctor makes absolutely no sense. We have a safe and effective vaccine that can prevent a lethal and morbid cancer. It's, from my point of view, shocking that we don't give this to everybody. So squamous cell carcinoma of the head and neck has largely been a surgical disease, and I'm trained as a head and neck surgeon, so I took care of a lot of these patients. And it's mostly because there have been very few drugs that have been proven to be safe and effective for this malignancy. And I will point out that the drug that is still most widely used, which is platinum, whether it be cisplatin or carboplatin, was approved way back in 1978. It's almost always given in combination with radiation therapy. It's a radiosensitizing agent. and it has quite well documented toxicities, which we don't need to belabor here today. 2006 was a banner year. We saw the FDA approval of docetaxel, which is not tolerated in combination with radiation and still used quite sparingly. And cetuximab, which is the first and only molecular targeting agent that's been approved for head and neck cancer. There was great hope in 2006 because of the ubiquitous overexpression of EGF receptor in human head and neck cancers, that this drug would provide a very effective alternative to toxic platinum chemotherapy. That turns out not to be the case. We completely lack predictive biomarkers, the level of EGFR expression in the patient's tumor, the level of phosphorylation of the receptor, amplification of the gene. None of these have proven to be adequate predictive biomarkers. And so today, cetuximab is largely given in combination with radiation in patients whose comorbid conditions mean they cannot tolerate cisplatin. But we, like everybody else in oncology, are now part of the immuno-oncology tsunami. And in 2016, we saw the approval of pembrolizumab and nivolumab, both immune checkpoint inhibitors blocking PD-1 for recurrent metastatic head and neck cancer. Pembro was approved in combination with chemoradiation in 2019 for locally advanced newly diagnosed cancer. And I would say that probably 90% of the clinical trials now are based on a backbone of immune checkpoint inhibitors. And so trying to figure out how a novel therapeutic strategy factors into the clinical armamentarium must take these drugs into account, which means for our preclinical models, we have to start working with immunocompetent models of squamous cell carcinoma to be able to test how these agents fit into future advances. So why should we all study head and neck cancer? And I don't think I'm speaking to an audience that needs to be converted, but unlike cancers like pancreas cancer or even lung cancer, colon cancer, head and neck cancer is very accessible for biopsy. We can do this under local anesthesia easily in the office. We have a fairly large number of well-characterized cell lines. They've been characterized by the Cancer Cell Line Encyclopedia, among other international and national efforts. It's not hard to develop patient-derived xenografts. We have nearly 100 in our lab, happy to share them, happy to I'll describe some data that we've generated using them. It's just important to remember that these contain murine stroma. We're finishing up a paper right now. I don't think I'll talk about it today, but we very carefully filtered out the mouse reads from our PDXs in order to mitigate the contamination of mouse stroma in the RNA-seq profile. And we found very, very little impact of doing that. So I think it's a liability. You certainly can't do immunotherapy studies because these models have to be grown in immunodeficient animals. But I think that their biology are largely reflective of the human tumor. We talked about HPV. You talked about how common oral cancer is in Southeast Asia. It's the most common cancer in India. Chemo prevention is feasible. When we see a patient with head and neck cancer, 50% of the time we'll cure them of their first tumor, but they will develop a second primary tumor at the rate of 3 to 6% a year, which means they are likely to die of another squamous cell carcinoma, whether it be of the head and neck, or the esophagus, or the lung. And the lack of predictive biomarkers is a theme, with the exception of PD-L1 expression as a predictive biomarker for the use of pembrolizumab and probably nivolumab. We really do lack predictive biomarkers to guide therapy for squamous cell carcinomas of the head and neck. So I had the privilege of collaborating with the group at the Broad at Harvard, led by Levi Garraway, Todd Golub, and Eric Lander. And we published this paper now about 10 years ago. And this was one of the papers that really highlighted the frequency of these notch mutations in human squamous cell carcinoma. And I look forward to the rest of the talks because it's really stunning to me that here we are 10 years later and we still don't know how to leverage this biological finding for therapeutic purposes. Notch inhibitors haven't made sense to test in these patients, and we still don't know what to make of the presence or absence of notch alterations with respect to treating patients. we decided to focus our energies, and you alluded to this, and I'm going to talk about this for the next few slides, on the most frequently mutated oncogene, which is the PIK3CA oncogene. I do want to point out that the mutations in HRAS are now coming to the fore as potentially really leverageable for therapeutic purposes. Many of you are familiar with the farnesyltransferase inhibitor story, particularly tipifarnib. These drugs were studied long ago. They were put on the shelf for lack of efficacy, like all of our prior studies. They were tested in unselected patient populations, but we now have some really tantalizing and compelling evidence that patients whose tumors have an HRAS mutation might be exquisitely sensitive to these drugs, and there's a phase 2 trial that hopefully will be completed soon in head and neck cancer to answer that particular question. So after we published this paper, we went back, we were doing the TCGA. I had the privilege of co-chairing the head and neck group, and we had amassed a large collection, and we noticed that the genes in this pathway, in addition to the oncogene PIK3CA, were by far the most commonly mutated genes in head and neck cancer that were potentially therapeutically leverageable. I'm not, P53, of course, is the 800-pound gorilla. I can't tell you how many P53 replacement gene therapy trials I've participated on, injecting P53 genes into human head and neck cancer for not very much benefit. So we decided to dive into this story a little bit deeper. And now with the publication of the TCGA, we have now a fairly large subset. So if we look at HPV negative squamous cell cancers in the head and neck that is not associated with the virus, 1/3 of these tumors will have a genetic alteration that activates PI3 kinase signaling. The majority are PIK3CA mutations. PTEN loss is also a common feature, which we studied as well. And if we look at HPV positive head and neck cancers, over half of those individuals will have a genetic alteration of the PIK3CA oncogene, making this now a fairly common genetic alteration and really amenable for precision medicine studies. So now back to the PDXs. Why am I a PDX enthusiast? Because it's so much easier to take a cell line and inoculate subcutaneously into mice. Well, here's what we found. Typically, as many of you know who do xenograft studies, the different models don't usually give us very different answers. Most cell lines are either sensitive or not to the drugs that we test in those models. But here we have a patient's tumor who has a canonical PIK3CA mutation, and we grew up this tumor, we made it into a xenograft, we implanted it into mice. And in this particular experiment, we treated it with an earlier drug, a combination mTOR PI3 kinase inhibitor. This went through clinical trials. It was stopped because of toxicity. But indeed, we find that we cured the mice with this drug. The tumors really disappeared. In contrast, when we treated mice bearing PDXs developed from stone cold wild type PIK3CA xenografts, they were insensitive. So it really convinced us that having a genetically defined model in the form of a PDX can actually illuminate the capacity of precision medicine. We then went back and we looked at the whole spectrum of PIK3CA mutations in human head and neck cancer. It looks very much like breast cancer and very different from other cancers. So for example, in colon cancer, all the mutations are canonical mutations. So we see that the three common canonical mutations in the helical domain and in the kinase domain occur in 2/3 of the cases. So it's still the most common PIK3CA genetic alteration in human head and neck cancer. But the story I'm now going to tell you that is unpublished and under review is a story about the non-canonical mutations. This is not a small subset. It's, over 1/3 of our patients have these rare mutations. And as all of you who participate in molecular tumor boards, when we get a targeted sequencing or whole exome sequencing on our patient's tumor, and we find that they have one of these mutations, nobody quite knows what to do with it. And as we saw from the FDA approval of the drug alpelisib in breast cancer, it's only approved in the case of canonical mutations or recurrent non-canonicals that have been proven to be activating. So we thought it was really worth characterizing these mutations in the context of head and neck cancer in anticipation of determining how to employ drugs that selectively target nodes in this pathway. So we've developed a model in our laboratory. In this model, it's a HPV negative squamous cell carcinoma model of the head and neck, and the parent line grows under low serum conditions. But we identified a clone and cultivated a clone of this model called SD1. And when we take away serum from the media of these cells, they begin to die. So in a very simplistic way, we can introduce a candidate driving oncogene into these cells. We can take serum away, and if the cells survive, we know that that gene is conferring a gain of function. Whether it's doing so directly or indirectly is a matter of a further debate, but it meets the criteria of a gain of function alteration. And so this is what our system looks like. We use a TET-inducible lentiviral vector that we flag tag to make sure that the transfected gene is into these cells. We turn on gene production, we serum starve them, and we test cell viability. And we come up with our answers. So I'm going to get right to the punchline. So here are all of the non-canonical PIK3CA mutations found in human head and neck cancer. This was a tour de force of a very talented postdoc in the lab, Nanxing, and what she found is that about 70% of these mutations are activating. And this is very similar to what's been found in other cancers where that's been studied. So it suggests that many of these mutations are actually worth paying attention to in our patients in terms of guiding therapy. And another thing that's really worth pointing out is it appears to be cell lineage independent. So my collaborator, Gordon Mills, who's now at Oregon Health Sciences Center, has a BAF3 model, a factor IL-3 dependent model, and he characterized all these same mutations with us and independently of us. There's also the breast model, MCSF10A. Again, these are factor dependent models, and you can see that a mutation that is activating in head and neck is generally activating in these other models, and in collaboration with some scientists in Israel, they have a HeLa platform, and HeLa is also a squamous cell carcinoma model, but in this case, it's HPV positive and it's cervical. And so there's a really large concordance across different cell lineages, which is, I think, very enticing and allows us to draw some conclusions. So now, what is the clinical significance of this? And finding this patient was actually extraordinarily hard, and I'll tell you why. We typically don't get mutational data on head and neck cancer patients, and there are very few studies that allow a patient to receive a P110 alpha PIK3CA inhibitor. But we had a postdoc in our lab from Korea. He knew about this ongoing basket trial in Korea of head and neck cancer, and we found such a patient. So here's an individual with a PIK3CA mutation. Nobody knew what this mutation meant. This mutation has never been reported in any human cancer, but he had a mutation, and by criteria of this trial, because this mutation was present in his tumor, he was eligible to receive the drug alpelisib. So he failed 3 cycles of chemoradiation therapy. He developed recurrence and metastasis. He failed 2 cycles of 5-FU platinum. And then he was put on alpelisib just because he had this PIK3CA mutation that nobody knew what it meant. And he managed to maintain, he responded, and he had a durable response for a year, and then he progressed, and he's now on durvalumab, an immune checkpoint inhibitor, and he's now 2 years later, and he's still alive. So this really shows you both the CT scan of his anterior mouth and lung metastasis, both of which reduced in size considerably on this P110 alpha inhibitor therapy, which is very encouraging. So here's a patient who's telling us that this mutation matters, and he got benefit from a fairly safe and effective drug for over a year. So we now started really focusing on this particular mutation. So back to our model, we induced wild type amplification, a canonical helical domain mutation, and this novel mutation in this patient's tumor. And indeed, each of these mutations induces phosphorylation of AKT. You can see it graphically here on densitometry, suggesting that this mutation turns on the PI3 kinase pathway. Also in our screening assay, it induced growth. So here's our control, and both the canonical mutation and this and now non-canonical mutation drive growth. So it does meet our criteria as a driving oncogene. We also are looking at other phenotypes in addition to survival in our serum-dependent assay. Here is an assay looking at colony formation, and indeed this novel mutation enhances colony formation. This novel mutation also enhances cell migration, very similar to a known canonical driving mutation. We did this with all of our mutation, and here and essentially found a very high degree of concordance between survival under low serum conditions, migration, and colony formation. But now we go back to our PDX collection. So we don't have a PDX that harbors the non-canonical mutation found in the patient, but we do have a PDX that has a non-canonical mutation that is shown to be a driver across all platforms. And indeed, when we implanted this PDX into mice and treated them with alpelisib, we saw very nice anti-tumor effects. In contrast, when we took a wild type PIK3CA PDX and we treated the animals with alpelisib, we saw very little impact. Interestingly, there is a pharmacodynamic impact of alpelisib in both settings, stronger in the activating mutation than in the wild type. But this really kind of closes the loop and suggests that from this day going forward, when our patients' tumors harbor mutations that have been shown to be activating, regardless of whether canonical or non-canonical, they should be candidates for treatment with drugs that target nodes of this pathway, including a P110 alpha inhibitor. And because alpelisib is now FDA approved in breast cancer, it's easier to get it for treatment in other cancers, at least in the United States. And this is just showing some colony formation with some other mutations, the patient mutation, this mutation of the PDX, and another mutation showing that we can drive colony formation that is blocked by alpelisib therapy. In contrast, these mutations that are all shown to be non-activating don't appear to form a lot of colonies, and there's no effect on colony formation by treatment with this drug. So it's a really nice and neat precision medicine story. This is complicated, but it's really where the biology is focused. So I'm not a structural biologist, but Natalia Yura is, and she's in the building right next to me. And we engaged in this really exciting collaboration where we were able to map the activating non-kinase domain mutations onto the known structures of PIK3CA, the non-activating non-kinase domain mutations, and the activating and non-activating kinase domain mutations. There's a lot of data here. I'm happy to share it with you as soon as this paper is published, but what I can tell you is that the structural mapping provides a really nice biological clue as to why some of these non-canonical mutations activate the pathway and others don't. In addition to looking at the structure-function relationships in our preclinical models and the structural biology, we are also doing protein-protein mapping. And this is a collaboration with Nevin Krogan and Daniel Swaney from his lab, and it's been a really enjoyable and productive collaboration. And then the basic idea is if we can map which proteins are associating with which other proteins in a particular cancer context, we might be able to design drugs that target an interacting protein when the protein that we're terribly interested in either doesn't have a drug that targets it or the available options are too toxic. So we did this with all of the PIK3CA protein mutations, and we came up with a PIK3CA interactome, if you will. And I'm going to tell you a really tantalizing story. This paper is in its second revision for a really high impact journal, so hopefully the story will be out soon. But what we found was really quite exciting. Specifically, we saw that the canonical PIK3CA mutations in the helical domain had a very strong interaction with several genes, IRS1 and HER3 in this case. We focused on HER3 because there are drugs that block HER3. We did co-immunoprecipitation and indeed found that these particular mutations do co-immunoprecipitate with HER3. And in contrast, and importantly, the other common canonical mutation, H1047R, which is in the kinase domain, does not strongly interact either by the interactome or by co-immunoprecipitation studies. So this would suggest that if a patient's tumor had one of these two mutations, it would respond to a HER3 inhibitor. In contrast, if it had H1047R, it would probably not respond to a HER3 inhibitor. And so we did that experiment. So in collaboration with Silvio Gutkind at UC San Diego, we're on this grant together. We engineered these cells and did this experiment. And I will call you, so we treated these drugs with a HER3 inhibitor that has been developed by Yossi Schlesinger and now licensed to Celldex. But this particular drug, so it's a Celldex drug, was highly effective in this head and neck cancer model engineered to express helical domain mutations. The tumors were very effectively growth inhibited. In contrast, tumors grown in mice engineered to express the kinase domain mutations were really insensitive to the drug, thus very neatly proving the hypothesis that this interaction between selected PIK3CA mutations and HER3 could reveal another strategy to target this gain-of-function oncogene. So in the beginning, when we first learned about this, we were really quick to design a therapeutic clinical trial containing this particular drug from Novartis, the P110 alpha inhibitor, BYL719, or now known as alpelisib. And then we learned that it was much more complicated, so we scaled back. And now in collaboration with my former colleague, but my collaborator, Julie Bauman at the University of Arizona, we are doing a window of opportunity trial. This is a platform that I love, helped to pioneer it. We've now put hundreds of patients on this kind of a platform. The reason I love this platform, it not only allows the surgeons to really meaningfully participate in research and thus really build robust translational research teams, but it allows us to get really important information about what a drug is doing in a particular patient in their tumor. So a patient is going to have resection. In this case, we've designed the study for oral HPV positive tumors because we know over half of them will have a PIK3CA alteration. They undergo a standard biopsy and we measure their tumor by CT scan. They then get 2 to 3 weeks of daily alpelisib, which is very well tolerated, and they have their surgery, in this case by transoral robotic resection. The paired CT scans provide a proxy of clinical activity, but we really can look at the impact of the drug on the immune system, on some of our candidate predictive biomarkers, and on HPV itself. We have about 10 patients enrolled on this study. We see responders, we see non-responders. We're in the process of immunoprofiling. We are making PDXs from these patients' tumors, and hopefully in the next year or so, we'll have a lot more to tell you about the impact of this drug in the setting of PIK3CA alteration or lack of PIK3CA alteration in human head and neck cancer. So the first part of my talk was a sort of a straightforward talk. I told you about a mutated oncogene. I talked about a drug that's FDA approved now that targets that oncogene itself and how we might use it. The next part of the talk for me is more exciting, but also more complicated because it essentially leverages our understanding about receptor crosstalk and receptor signaling and how a mutated oncogene could have implications for other drugs to be brought into the clinical armamentarium, ideally in the setting of prevention, that would really make an impact for our patients. So you may be familiar with this 2012 paper from the New England Journal. It's based on the Nurses' Health Study, and this is straight epidemiology. They looked at this very large cohort of patients. They knew who had colon cancer. They knew what their survival was. They knew if they used aspirin, which is a fairly well-accepted chemoprevention for colon cancer, and they had the tumor genetic information. And it was surprising that they found that patients whose colon cancers had a PIK3CA mutation, and remember I told you these are all canonical mutations, they had a significantly improved survival if they used aspirin. So we thought, based on our receptor signaling work over the years, that we knew a lot about this, and potentially there was a mechanism to leverage this information to help patients with squamous cell carcinomas, in this case of the head and neck. So we heard earlier about ADAM17, G protein couple receptors can transactivate tyrosine kinase receptors, including EGF receptor. Part of that transactivation pathway critically flows through a PIK3CA. We also know that PI3 kinase is activated downstream of even just wild type receptor tyrosine kinase signaling. We'd studied crosstalk. We knew about the prostaglandin E2 receptor, and we knew that NSAIDs had been looked at in human head and neck cancer, but without a deep understanding of biology, and usually in combination with chemotherapy. And the results were not very informative or tantalizing. So we decided we would just test the hypothesis once and for all. And the beauty of building a center the way you are is you're going to have high quality tissue. You're going to have phenotypic information on the patients who provide the tissue. And if you also collect the information about what medications they take, either prescribed or non-prescribed, and the reasons that they do this, you will have the ability to ask this question, which very few people do, which is why our study kind of came out of the blue, because there was no other cohort that had all this information. So we had a large tissue collection. We knew whether or not the patients were on chronic NSAIDs. We were able to sequence PIK3CA. We also did FISH to look at gene amplification, which we also think is important. We looked at HPV, and at the final case, we had 266 patients on whom we had overall information. And it was a little bit of a Hail Mary. We asked the very simple question: if you're a head and neck cancer patient and your tumor has a PIK3CA mutation, and you happen to be taking NSAIDs, and in the vast majority of cases, it was baby aspirin for cardiovascular reasons, did you have any change in your overall or disease-specific survival? So here's the answer. It was pretty stunning. When we looked at our patients' tumors who were wild type without amplification, it didn't matter if they never used or chronically used NSAIDs. Their survival at 5 years was exactly what we expect. It was about 45, 50%, and there was no difference in whether and survival. And this is, I'm showing you disease-specific survival because it's more rigorous. The same thing was true for overall survival. However, if your tumor had a mutation or amplification in PIK3CA, your survival was worse. That's not surprising. That's been shown in other cancers. This is an oncogene. However, if you had the luck to have a mutated or amplified oncogene and be on a non-steroidal anti-inflammatory drug for at least 6 months after your cancer was diagnosed, your survival at 5 years goes up to almost 80%. This was independent of HPV, it was independent of other clinical and pathologic variables, and it was really quite remarkable. So we then went to our preclinical models, which are really challenging to recapitulate what you find in patients, but we think we found some answers. We had a whole suite of PIK3CA mutant PDXs. I showed you some of that data earlier, and we treated these mice with vehicle or an NSAID. We did this with Sulindac, indomethacin, celecoxib, and aspirin. We found the same thing in every case. I'm showing you the Sulindac data. So in the case of a wild type PIK3CA PDX, it's insensitive to NSAIDs. That's not a surprise. NSAIDs should not cure cancer. However, we were able to show that if it had a mutation in PIK3CA, it grew quicker. And if you treated these animals with an NSAID, you had significant anti-tumor effects, and that was pretty remarkable. That was surprising. We then asked the question why, and we really thought it had something to do with prostaglandin E2, and we think it has something to do with the immune system, because remember, the patients are always telling us the truth, and we found in our cohort that they were disease-free until their disease recurred. And it was the mutation in their tumor that ultimately dictated this pattern of recurrence and survival. So we hypothesized that it had to do with some potent circulating factors that could interface with the immune system. Prostaglandin E2 was logical. So we then measure prostaglandin E2 levels in the circulation of mice with PIK3CA PDXs. Here are individual mice with wild type PIK3CA PDXs and individual mice with mutant PIK3CA PDXs. And we see a significant upregulation in the blood of the mice who are implanted with patient-derived xenografts, either wild type or mutant for PIK3CA. I then mentioned the immune system. We then engineered some models, and now we're growing them in immunocompetent mice. These are MOC1 cells that are very well characterized and known to respond to some degree to immune checkpoint inhibitor. And indeed, when we engineered the cells to express high levels of PIK3CA or this canonical mutation, we saw a significant decrease in NK cells in the tumor. And we saw a similar decrease in CD8 positive T cells. So we now think there's some potential immune mechanism to explore in patients that we're going to test in our patients on that the ongoing window trial. But the definitive trial really needs to be an adjuvant trial where we cure patients of their head and neck cancer. We have the genetic information about their tumor, and they're randomized to receive NSAID or placebo for 2 years. We are modifying this slightly. We have there's some really interesting data coming out of the colon cancer world. They did a study like this in colon cancer, but they were, for various reasons, compelled to use celecoxib. They found no impact on celecoxib. We don't think that there's any evidence that it's COX-2 specific, so we would not want to use celecoxib. And we think there's a lot of good reason actually to use full dose aspirin as opposed to baby aspirin. And so we're modifying the trial design and looking for a possible way to fund this trial, because without a pharmaceutical supporter, it becomes a really challenging economic enterprise. So I know it's tempting to say, oh, you've already shown it in a retrospective cohort. Can't you just take a baby aspirin or an aspirin a day if your tumor has a PIK3CA mutation? Sure, but all of the really compelling data show us that aspirin is not without toxic side effects, particularly bleeding, particularly in elderly patients who have no compelling indication to take it. So I'm hopeful that someday someone will do the right prospective study and definitively prove whether the presence of a PIK3CA alteration in a squamous cell carcinoma is actually a predictive biomarker for disease prevention and prevention of recurrence and new primary tumor formation with aspirin. And that would be sort of a major benefit to people for all the right reasons. So in the last few minutes, I'm going to switch gears. PIK3CA is an oncogene. They're drugs that target it directly or indirectly, but transcription factors are much, much harder to target. So I'm going to tell you a little bit of story about a driving oncogene that is STAT3 in human squamous cell carcinoma. So STAT3 is a very attractive cancer target for 2 reasons. It's overexpressed and hyperactivated in the majority of solid and hematopoietic cancers by the tumor cells. So it has a phenotype on the tumor cells, but in this era of immuno-oncology, it is also hyperactivated in tumor infiltrating immune cells where it is immunosuppressive. So if we actually had a drug that could block STAT3 in the tumor, we could enhance anti-tumor immunity and increase tumor growth, and that would be quite a home run. So adding fuel to the fire was this paper from Jeff Settleman when he was at Genentech that didn't go in to find STAT3. He was really looking at drug resistance mechanisms in the setting of tyrosine kinase addicted cancer cells, and found STAT3 that emerged as the primary mechanism of drug resistance, which is actually how we got to it in head and neck cancer. It was in our patients who were resistant to cetuximab. It was hyperactivated, and hence we started to study it. Now, the mantra is that transcription factors are undruggable, but I hope in the next few minutes to convince you that that's not entirely true. So Most folks have tried to block downstream signaling using upstream inhibition of either receptor tyrosine kinases or non-receptor tyrosine kinases. We've looked at all of these strategies. They work, but there's a rapid emergence of drug resistance, and it's not the most effective STAT3 inhibitor. I won't show you any of that data because I want to get right to our STAT3 inhibitor story. So being a naive surgeon, I created a drug. I don't know if I had known what I know now about drug development, if I would have done this, but it seemed like the right thing to do. We'd read a paper about a decoy blocking STAT6. It was not difficult to synthesize or design the drug. We very carefully made it as small as possible, so it's considered a large small molecule. We were able to show that it bound to STAT3 dimers in the cytoplasm. It crosses into the nucleus. It binds to the promoter regions of STAT target genes. And this kind of drug is really a first in class, and there's no really plausible mechanism for drug resistance, and we haven't seen drug resistance. So our first generation molecule that we took through to a phase 0 clinical trial looks like this. We had phosphorothioate modifications on each end. In our preclinical work, we have a mutant control version that just differs by a single base pair, but does not bind to the STAT3 response elements in the promoter genes. We and many, many other people have studied this. Its significant limitation is it has to be injected into the tumor because it's not stable in the circulation. So this is what we did. We did a clinical trial. I took my patients to the operating room. I randomized them. We biopsied them in the operating room. Half the time we injected the tumor with our STAT3 decoy. Half the time we injected the tumor with saline. And then anywhere from 2 to 6 hours later, after the neck dissections were complete or the flap was in place, we took out the tumor. And it was a very simple question, pharmacodynamic. If we inject the decoy into the tumor compared with saline, do we see evidence of downregulation of STAT3 target gene expression? And the answer was yes. So we chose, in this case, BCLXL and cyclin D1. There's just 2 of the many STAT3 target genes. And you can see that in these 4 patients who got the decoy, there was lower expression following decoy therapy with a very brief exposure. We did immunohistochemistry on the large cohort, and this was 15 patients got decoy, 15 got saline, and we found this significant difference, and we published this. But I was done with an intratumoral formulation. So with the collaboration of a chemist who specializes in nucleic acid therapy, Daneth Lee at Carnegie Mellon University in Pittsburgh, we went through many iterations, but we ended up with our decoy stabilized by the addition of these carbon spacers on the ends. And now this is really a drug that can be given intravenously. So we were able to show that there's really a nice dose response in a xenograft model. And we've experimented with the frequency of injection. What I can tell you very confidently is you get the same anti-tumor efficacy with twice-a-week injection as you do with daily injection, and we're now exploring once-a-week injection. And we find no benefit to going above 5 mg per kg of the decoys. And these are all tail vein injections in these mice. A reviewer asked us to prove that it was better than an FDA-approved drug that blocks STAT3, in this case ruxolitinib, and indeed here's ruxolitinib and here's our decoy down below, so it is. And they asked us, how does it compare to cetuximab? Cetuximab's pretty good in the xenograft model. Our decoy was a little better, and indeed when we combined the two, the tumors disappeared. So that was encouraging in terms of future translation. But now, of course, it's the era of immune checkpoint inhibitors. So this is our first experiment using an immunocompetent squamous cell carcinoma model. These were the MOC1 cells. We combined our decoy with the murine version of anti-PD-1, and indeed we see that we can get a significant improvement in percent Treg downregulation when we give the decoy in combination with the checkpoint inhibitors compared to our mutant control decoy and the checkpoint inhibitors. So we're continuing these studies and looking at the immunoprofile of this approach. In addition to downregulating those aspects of the immune system, we increase interferon gamma expression with the combination of the decoy and anti-PD-1. So we're hopeful that's the way to go. But I want to finish by telling you that it's not just squamous cell of the head and neck. Here's a study in a squamous cell of the lung by my colleague Jill Siegfried. This has been published. She saw anti-tumor efficacy of the cyclic STAT3 decoy in her models of EGFR mutant tyrosine kinase inhibitor resistant non-small cell lung cancer. But maybe the most tantalizing data is the following. And this came from her graduate student who decided when he was doing a carcinogenesis experiment with NNK just to see if the decoy had any impact on tumor outgrowth. But he didn't remember that he'd treated the mice with the decoy, and these take months and months for the tumors to outgrow. And one day Joe gets a call that she's got a cage of mice that can't breathe and a cage of mice that are perfectly fine. Can she come take care of them? And the mice that were perfectly fine were the cyclic STAT3 decoy mice, and the mice that had lungs filled with lung cancer were the cyclic STAT3 mutant mice. So they'd gotten the inert control. And so we decided to expand on this work. It's now been published, but we are taking this idea of prevention. The NCI is now funded through its Prevent Network, creating an aerosolized version of the STAT3 decoy. So Jill is now working on that. And the idea is that you could inhale this to prevent conditions that are dependent on STAT3, including lung cancer, and now really interestingly, probably COVID-19. So lots of applications with the inhalation format, just early days of starting that. But the final piece that I want to close on is the opportunity to do clinical trials in patients who aren't people. And my sister is a veterinarian, and she has regaled me for years with the tragedy of cat oral cancer. So pet cats get oral squamous cell carcinomas, and they all die. They get radiation. The radiation isn't effective. Their oral cavities are too small to tolerate the excision. And so we looked at, in collaboration with veterinary pathologists at the University of California, Davis, which is just about an hour away from San Francisco. And indeed, STAT3 was hyperactivated in about half of those tumors. And when they look at the tumor microenvironment of their cats with oral cancer, it looks very, very much like human head and neck squamous cell carcinoma. So we now are about to open a clinical trial in pet cats. We were able to fund this with an R01 because unlike a clinical trial in people, you just need IACUC approval. You don't need an IND. So the manufacturer is ongoing and we hope to open this at UC Davis in the next few months, but it's going to be a phase 1-2 trial of the cyclic STAT3 decoy to pet cats. And even though reviewers keep forgetting that these are not laboratory animals, I keep reminding people that we can't do things to these animals like we do in our laboratories because these are members of people's families. But we are going to access the tumor and the tumor microenvironment and hopefully get a lot of information that could help us when we're ready to roll out this treatment for patients, human patients with squamous cell carcinomas. So I'd like to conclude by thanking you for inviting me. I only wish I could be there. Please invite me again when you're open for business. I'd love to come. But I think in cancer research, we really are at a confluence of a deep understanding of biology and genomics, and we really have to grab the bull by the horn, identify predictive biomarkers, even if it's hard. So there great opportunities. The challenges are immunotherapy is here to stay. We have to contend with that in our models and in our new treatments. And I just want to emphasize the incredible importance of tissue biopsies on treatment, particularly in the setting of resistance to identify underlying mechanisms. And I want to end by thanking all of my collaborators and funding sources at UCSF and elsewhere. And thank you very much. I'm happy to stop sharing my screen and take questions.
Jennifer, that was absolutely superb, very inspiring.
Oh, thank you.
And there's so many things going on there, but all sound all positive. You know, lots of nice therapeutic approaches going there. And as somebody who likes doing human genetics, showing the value of sequencing the tumor to actually think about targeted therapy. So I think, you know, I don't know how many people, how many hospitals are able to do that, but I think it's certainly in head and neck cancer. It sounds like it's certainly should test for the PIK3CA mutation anyway. Yeah. So so can I start off asking a question about all these JAK inhibitors that are coming out at the moment?
Oh, absolutely.
Because they're all, I mean, I don't know much about them, but they're all being developed for treating inflammatory disease like psoriasis and all sorts of things. But you say that they would work initially in the patients, but then they start working because they get resistance. Is that right?
Yeah, well, that would be the hypothesis. So we have some experience. So I will tell you that we are doing a clinical trial now with ruxolitinib. It's a window of opportunity trial. We have given ruxolitinib prior to surgery to about 20 patients. The best clinical response we've seen is stable disease. We have not seen any tumor shrink in that 2 to 3 week period. So I'm really skeptical that it's not going to be a meaningful agent, certainly as a single agent. But what's fascinating about the data, and again, this is hot off the press, so I wasn't sure I could really present it yet, is we made PDXs from these patients' tumors. And we took the PDX from the patients who had stable disease. And indeed, there was some evidence of an anti-tumor response in the animal. And we saw downregulation of phospho-STAT3. In contrast, we made PDXs from patients whose tumors progressed on the therapy. And we did not see downregulation of phospho-STAT3. So in that particular case, it could be that there is something about the biology of these drugs and access to the tumors. And I don't really understand, enough about, pharmacodynamics and drug delivery with an oral agent, because these are all oral agents, but they're clearly not going to be a home run. We've also looked at tocilizumab, which is the IL-6 antibody. We've only done it in preclinical settings, but we've really seen very little evidence of activity. So we haven't been moved to go in that direction. So I guess my hypothesis of all of these agents is that the reason STAT3 is activated is as a result of multiple positive and negative regulators. So there's activation of upstream kinases, which is what we focused on. The story I published previously, and I didn't talk about today, is I think the main reason that STAT3 is activated is loss of function of the negative regulators, which is these protein tyrosine phosphatases, namely PTPRT and PTPRD. And it's really a devil to restore function of inactive phosphatases. So I think that, frankly, if we can get a good STAT3 inhibitor, that's the way to go, because otherwise you have these competing forces of activating and inactivating alterations that ultimately allow the tumor cells to survive just knocking out one of the upstream activators.
Yeah. We've got quite a few questions coming through. I'm just going to go through them as I see them. So is this a question from Mavash, is STAT3 hyperactivation and hence targeting specific to HPV negative head and neck cancer? It is not.
It is not. STAT3 is rarely mutated in any cancer, only a subset of T-cell leukemias. But we have not been able to distinguish a characteristic cohort that would be uniquely susceptible to the drug. So we've looked at STAT3 null models, and of course in STAT3 models they don't respond to STAT3 inhibitors, but it spans HPV positive, HPV negative, and just like with the EGFR story, we have no evidence that the level of expression of tyrosine phosphorylated STAT3 or STAT3 activation has anything to do with whether or not the tumor will respond to a STAT3 inhibitor.
Brilliant. Lots of questions coming through and lots of nice comments saying what a great talk it was and easy to follow. Question from Yung Wang is there are a portion of SCC patients who have no detectable mutations in PI3KCA, but the gene is highly amplified. Would they also benefit from this therapy?
Yes, we think so. So we've looked at amplified and we've looked at mutated, and we do see a signal in amplification. So the amplification almost always gives rise to pathway activation. There may be some really important but subtle differences between which drug and therapeutic approach should be deployed for which mutation or in the setting of amplification, but we think actually gene amplification matters.
Okay, great. Can I go back to this HPV?
Sure.
About the poor take-up or the rollout for some people. Do you think post-COVID, where obviously countries have had to develop vaccination strategies and people are used to taking vaccines now, do you think there'll be a greater take-up by by parents allowing their young boys to actually have the injection? The virus?
You know, it's a great question, David. I hope so. You know, it was very interesting. I was just interviewed last week by a group of high school students who learned about HPV cancers and were appalled that their parents were not allowing them to be vaccinated. And they went around interviewing people, and I had the chance to learn from them what the anti-vaxxing mothers say. So they actually, you know, Marin County, which is north of San Francisco, one of the wealthiest counties in the world, has a very high prevalence of anti-vaxxers. And what they shared with me is, you know, from my point of view, it's usually not trusting the medical system, but that's not the case for wealthy white people. And what they found is that the need to control the health of your child, what your child puts into their body, what the child does with their body, is something that is much more important to some parents and to others. And I think that, quite frankly, just as we've found ways to liberate children from relying upon their parents to grant permission for, frankly, terminating pregnancies, we need to be able to go right to the susceptible population, educate them about what this vaccine will do for them throughout the course of their life, and enable them to get vaccinated without relying on their parents. We don't see any vaccine hesitancy among teenagers. It's all among their parents. So in addition to, you know, hopefully COVID saving your life and these new vaccination strategies, hopefully there'll be an empowerment of the susceptible population who, you know, when they learn that their parents haven't vaccinated them, they're just stunned.
Yeah, amazing. I think we're going to have to close there. Hopefully you can perhaps answer some of the questions when you get time on
Sure.
that are in the FAQs. That was absolutely superb. And thank you very much for that.
Thank you very much for having me. I hope to meet you in person someday.
As soon as you're allowed to travel, please come to the Blizzard.
I would love to. Bye-bye. Take care.
Bye-bye.
I'll stay on.
Thank you. Bye. Okay, I'll pass you on to David Williams, who will chair the next session.
David, thank you very much indeed. I don't know whether you can see me at this point or not.
See you, David. You're in the mountains.
I'm actually in Yosemite Valley in California, so not very far from San Francisco. It's absolutely fascinating this afternoon to reflect on the quality of this symposium. When I was Deputy Vice Principal for Health some years ago, we were commissioning an architect to build the Blizzard Institute. One of the guys who presented himself was Will Alsop, who was slightly left field compared with the other architects who were proposing plans. And the rest of the committee turned to me and said, well, what do you think? And I said, you know, I think if you don't give the academics at Queen Mary a chance to work in a building like this, they'll never forgive you. And they were persuaded by that. And I have to say, it's a cause of real pleasure and satisfaction from my point of view to see the success of the building and the quality of the research coming from it. And this afternoon is just one great example of that, and I think really promising for the future of the centre. So with that said, it's my pleasure now to introduce you to Dr. Manu Mathur. He's a dentist. He's a PhD in epidemiology and population health, which he gained at UCL. And that's when I first met Manu. When I was his external PhD examiner. He's currently in the Department of Dental Public Health at the University of Liverpool, and also holds the position of Head of Health Policy and additional professor at the Public Health Foundation of India in New Delhi. In addition to that, he is also a commissioner on the recently constituted Lancet Commission on Global Oral Health, and is going to look at oral cancer now from the whole animal perspective, if I can call it that. And it's now, Manu, my pleasure to invite you to give your talk on smokeless tobacco and oral cancer, a burgeoning epidemic, the low and middle income country perspective. Manu.
Thank you, Professor Williams, and good afternoon. Hello, everyone. It's a pleasure to speak in this symposium, and as Professor Williams mentioned, it's such an illuminating experience to listen to all the fantastic talks which happened before me. I would take a slightly different angle to my talk. We heard about all the clinical side of things. I would talk from more from an epidemiological point of view on a substance which is one of the major causes of oral cancer, and it is often ignored. And it is delightful when Professor Coulthard opened his presentation this afternoon, saying that PAN is one of the major causes of oral cancer. Well, exactly that. This is the problem, that it is PAN which acts as a carrier to many other things, which constitutes smokeless tobacco. And it is an often ignored thing. It is often overshadowed by the smoking practices and by the mortality and morbidity which happens due to smoking. So just to start off my presentation, I would just like to just take you through the global burden of tobacco use. It kills up to half of its user. It kills more than 8 million people each year, 7 million by direct use and 1.2 million by the indirect effects of secondhand smoke. Just to summarize, there is not even a single organ in our body which is not affected by tobacco use. So starting from stroke in the brain, going down to gangrene in legs, every possible organ in the body is affected by tobacco use. And from an epidemiological perspective, if you look at what is the burden, what is the attributable burden on disability adjusted life years of non-communicable diseases, tobacco is the third largest contributor to disability adjusted life years. And it is the second largest contributor if we just take the male population. If we do a further cross-section of this, the highest contribution to DALYs for NCDs is in Central European and East European countries, followed by the Southeast Asia and East Asia countries, and then the high-income countries like United Kingdom and United States of America. So you can understand what kind of burden the habit of tobacco causes. Now, when we talk about tobacco, we actually talk about smoking forms of tobacco a lot. Whereas if we talk in terms of from an oral cancer perspective, it is the smokeless tobacco which is even more harmful. And it took us many years actually to correctly define what exactly we mean when we say smokeless tobacco. So this is a definition which we gave in the monograph, which was commissioned by World Health Organization in 2016, which said that all commercial and non-commercial products that contain tobacco and are used without ignition at the time of their consumption, and they are either consumed orally or nasally, mixed with various sweetening agents like spices, areca nut, lime, etc. And they have the same kind of addiction potential like smoking forms of tobacco. What is even more dangerous is that it leads to formation of nicotine depots in your oral cavity, which provides a sustained release of nicotine, which makes it even more addictive and even more harmful. So, as has been said in various presentations before me, smokeless tobacco consumption is mainly in the South and the Southeast Asian region. But again, this is a misnomer, and there was a global report on smokeless tobacco which was published a few years back, and which said that the consumption is prevalent in about 127 countries of the world. There are about 356 million people who consume smokeless tobacco right now, and it contributes about 8.6 million disability adjusted life years and about 350,000 deaths a year. And if you can see on the right side of the screen, there are various kinds of smokeless tobacco products which are prevalent. So it's not only the pan, which is the upper left image. Pan historically has been used in South Asia as a roughage. It is good for digestion. So pan on its own is not very harmful. It is the areca nut and the smokeless tobacco which is added into pan which makes it harmful. And because it is so much culturally ingrained in the societies, there is no social taboo like smoking. So it can easily be consumed anywhere. And if you see in the second row on the right, this is something which is prevalent in the northeastern parts of India and in Burma. So what they do is they take tobacco leaves. So if you can see my background, this is the border between India and Myanmar. And if you can see the greenery around me, these are all tobacco plantations. So the villagers, they take these tobacco leaves and they just smoke it and pass it through water until it becomes orange in color. And that is served in marriages. So it is very much ingrained in culture, and it's not only South Asia. If you see the lower right picture, this is a picture of khat, which is very common in sub-Saharan African countries, and it is sold in the market just like tea. So it is very much prevalent in South Asian countries, but it is seen everywhere in the world. And this is the reason why smokeless tobacco is often ignored when we talk about epidemiology, when we talk about the public health burden. This is a picture which I've taken from the Global Burden of Disease study. And if you see, this is the distribution of smokeless tobacco users. Now, there are so many smokeless tobacco users in South Asian countries like India, Pakistan, Bangladesh, and Myanmar, that everything else is overshadowed. Everything looks dark blue and light blue, and it looks like that it's not a problem of the world. It's not a global public health problem. It's just a problem of the subcontinent. I'll come to that picture again. Just giving you, I don't need to go into details about this because we have heard since morning, it contains about 3,095 chemicals. 36 of them are carcinogenic. It is a major cause of esophageal cancer, oropharyngeal cancers, oral submucous fibrosis, and many other diseases. And there are studies which have been done in various parts of the world which have actually shown this kind of association between smokeless tobacco use and different kinds of cancer. In terms of oral cavity, the risk of oral cancer is 2.6 times greater for people who consume smokeless tobacco. It causes recession of gums, it causes tooth loss, it causes permanently stained teeth, we know this. And another important constituent of smokeless tobacco, areca nut, is often associated with oral submucous fibrosis, which is restricted opening of oral cavity or a precancerous lesion. Now, the place where I did my dentistry from, I used to get at least 4 cases of oral submucous fibrosis in a day. And this was huge amount, and they were all smokeless tobacco consumers, all gutka consumers. So talking specifically about smokeless tobacco and oral cancer, and we're talking in the Squamous Barts Centre for Squamous Carcinoma. We have talked about this. Yes, the nicotine and the other products are released in the body as carcinogen. They are metabolically activated, form DNA adduct, leads to gene mutation and cause cancer. This is the modified HEC model. And I think speakers before me have elaborated on this a lot. But look at the amount of chemicals which are there in smokeless tobacco. So it's not only the tobacco-specific nitrosamines which are causing the problem. It contains polonium, which is a new radioactive material. So the manufacturing of tobacco leads to release of polycyclic aromatic hydrocarbons. Now, these are also carcinogenic, and there are various harmful metals which are added to it, like arsenic, beryllium, cadmium, chromium. And all these substances are the ones which are cause cancer. And these are all documented in the IARC monograph on cancer. So these are all the substances which are there. I can go into details, and I think I'll be talking to experts if I go into detail of the mechanisms. But coming back to this picture again, why are we talking about smokeless tobacco when we are talking about oral cancer? And why am I stressing on smokeless tobacco? Well, yes, the burden is in South Asia, but it is increasingly becoming prevalent all over the world. In the 1950s and 60s, before Dolinil gave the classic study on tobacco and smoking and lung cancer, tobacco was also considered as just a product, a commercial product, which does not cause anything. But slowly it became a movement. People started realizing that it is spread like an epidemic. And this is happening exactly the same way. It is following the same science of the smoking epidemic. Smokeless tobacco is following the same science. That is why it is important to understand that this is not a correct representation of the world. So let me just talk about the current data on cancer risk in smokeless tobacco. And what are the challenges in front of us? And how can we, what are the challenges when we're trying to decipher the evidence, when we try to tease out the evidence between the links between smokeless tobacco and cancers? We did a review of the reviews, and we published this paper in the Asia Pacific Journal of Cancer Prevention. I was one of the authors of this. And we actually looked at studies with the population or hospital or mixed studies which have actually looked at the relationship between cancer mortality and smokeless tobacco use. And we tried to tease out what can be the challenges which helps to prevent a very robust association between epidemiological association between cancer mortality and tobacco use. And we found there are 5 challenges. And the first challenge is to consider smokeless tobacco as a homogeneous product. Unlike cigarette, smokeless tobacco is not a homogeneous product. in every country of the world, it is present in a very different kind of form with a very different kind of constituents. And it's not only South Asia, every possible continent in the world have their own local variant. So for example, chemo or rape in South America, talking about loose leaf chew or eat meat in the Northern American countries, tumba, khat, dry snuff in African countries, shama in Middle East, snus, which is very common in Sweden and Norway, and then naswar, which is taken through the nose in Pakistan. India, we have many other types, mainly betel quid with tobacco, as I said, Dahra, Kenny, many. And then Bangladesh, smokeless tobacco is used as a toothpaste, in eastern parts and in many of the South Asian countries. So it's not a homogeneous product. It's a product which has got different constituents, and we have to look into different constituents of different products in order to really understand what can be the burden of this product on cancer. Then the second challenge is defining the patterns of smokeless tobacco use. Now, if you read through the Global Adult Tobacco Survey, or if you read through the WHO papers, there are very clear-cut definitions which have been used in order to define smoking forms or smoking. Even for e-cigarettes, there is a very, very, very elaborate module. But when you start looking into studies which actually measures the burden of smokeless tobacco or try to find out association between smokeless tobacco and cancer, there are about 55 different definitions on smokeless tobacco use. And these are some of the examples which we were able to find in our review. So how on earth are you going to collate this evidence? How on earth are you going to make sense out of this evidence when we have not really defined how can we measure the patterns of smokeless tobacco use? How are we defining current use? How are we defining level use and common use of smokeless tobacco? Now, the third challenge, and I think this is a very important challenge, is the limitations of study in females. As I said, it's a very, very cultural thing. And again, going back to the initial presentations, even if you see in East London, this is a habit which is particularly prevalent amongst females. Because it is culturally acceptable. You can keep a betel quid, or you can keep a quid of gutka or canny in your mouth, and you can pass the whole day. It gives you consistent supply of nicotine, which keeps you euphoric, and you can work throughout the day. And nobody is going to observe you, or nobody is going to judge you. But people who are the most prevalent users of smokeless tobacco are the ones who are studied the least. So there are only 11% studies, population-based studies, which are actually looking at the association between smokeless tobacco and oral cancer. There are only 10% hospital-based studies. So clearly there is a mismatch there. Clearly we are lacking in evidence at many places. Then the fourth challenge is paucity of good population-based studies. We require good population-based studies in order to establish this association. And you can see that what I have done here is I've just looked at studies which have tried to establish a link between smokeless tobacco use and oral cancer. So only 31% studies are population-based. And even in these studies, smokeless tobacco has been a very secondary outcome. So it's a question which has been asked in passing. So do you consume smokeless tobacco? That's the only question. No defining on what are the things which are being included in smokeless tobacco. Nothing of that sort. It's just a passing question, which is asked with 8 to 10 questions on smoking and 3 to 4 questions on e-cigarette. So how on earth are we going to generate evidence around this problem of having very less population-based studies? And those which are there are very weak. And this is a huge challenge with us. Then there is a geographical limitation in evidence. Although tobacco is consumed in 127 countries of the world, but I would like you to go to the Smokeless Tobacco and Public Health monograph, the global perspective. And if you look at the evidence on oral cancer, there are only 25 studies which actually finds out the link between oral cancer and smokeless tobacco use. And most of these studies are either in South Asia or in Western Europe basically meaning Sweden and Norway, and from America, 1 or 2 studies from America. Now, how are we going to get this evidence? How are we going to establish the burden or establish a robust linkage when there is so much geographical limitation in the evidence? So just to sum up, 20th century was all about shared vulnerability. We were talking about how vulnerable we are, and global health was talked about because we were talking about these vulnerabilities of HIV epidemic and many other epidemics. I think 21st century, I use this slide and I say that 21st century is a century of shared values. I have added vulnerability again because COVID pandemic has actually made the entire world more vulnerable. And so is the case with smokeless tobacco also, because with increasing trade and with increasing availability, if I go on amazon.co.uk right now, I can buy smokeless tobacco product. Nobody will ask me whether I'm 18 years or 20 years old or not. Like if you go in an off-license shop and buy a cigarette. So yes, 21st century is all about shared values as well as vulnerability. Universal is local without walls. And so we have to learn from each other. We have to learn from the experiences, and we have to see an epidemic surfacing somewhere in the world and have to be prepared for it. So is smokeless tobacco really a public health issue? Yes, I think it is. And I think you would agree with me that it is. It has got health effects. It causes a lot of cancers and pre-cancers, leads to addiction. Increasingly, there is a set of researchers in UK, in US, and many parts of the world which actually promote smokeless tobacco as a harm reduction measure. So there is this whole issue that we have a lobby which says that smokeless tobacco is a harm reduction tool, and there is dual use. So people who are not able to consume cigarette, they will resort to smokeless tobacco, and they will resort back to cigarette whenever it is required. There is an economic burden due to healthcare costs, due to cost of consumption of tobacco product, due to heavier investment in tobacco farming. There is an environmental impact of smokeless tobacco because of infection from the spit, defacing of public places, degradation of the soil layer because of the cultivation. And of course, there are socio-demographic issues associated with smokeless tobacco. As I said, it is deeply embedded in customs. It is highly consumed by women, leads to child labor. And if we talk about cessation practices and measurement tools, we are doing very, very poorly. So even if you talk about cessation, if you talk about looking at scales and tools which measure addiction, they are mainly made around smoking forms of tobacco. There is very less tools which actually talk about cessation of smokeless tobacco. So we have to work on that. This is just to show that very little research is done on smokeless tobacco in UK. This is just an example from the WHO Empower report. You can see that the smokeless tobacco columns are blank. This is another one from the FCTC report. Again, smokeless tobacco columns are blank for UK. And this is from the global burden of disease. These are all skewed values and hard to believe values. So we need to develop this critical mass of research. Now, when we talk about research, and I'll tell you why I'm talking about this slide. Every research is done so that it is converted into some kind of a policy. Knowledge has to turn into action. And knowledge can only turn into action when there is interdisciplinary research which is involved. So you need to establish scientific credibility. We need to establish evidence and rationale. And for that, we need biomedical and epidemiological research. We have heard a lot of fantastic studies this afternoon. We need to ensure financial feasibility, whether the interventions which we are promoting or the intervention which we are proposing are cost effective and affordable. We need operational stability, whether the things which we are proposing are sustainable and scalable, whether the treatments we are proposing, whether the interventions we are proposing are sustainable and scalable. And the most important thing is political viability, whether the community is ready and receptive for the kind of intervention which we are proposing. And for that, we need social science research. One size will not fit all. We have to ensure that the communities are ready and receptive for this. So I was looking through the Barts Centre for Squamous Carcinoma webpage, and I think there are tremendous opportunities which can be utilized by the center. And especially because of the location where you are located and the kind of research which has already been done and which has been put into. There are four dimensions in which research and in which various research problems can be looked into. And epidemiological research, we can talk, this whole thing around big data and data linkages. We have to fill out these critical gaps in data which are present. because things which get measured, measured are the things which get funded. If things are not measured, they will not be funded, they will never be converted into policy. So we need to get that data linkages in. We need to understand what are the comorbidities which are associated with oral cancer. We have to work around the treatment efficacy for various kinds of treatment for oral cancer, looking at tumor response, disease progression, and reoccurrence. We need to establish effective social science research. So what can be the effective health promotion program? What are the area level inequalities which are present? And what is the impact of oral cancer on quality of life? Until and unless any intervention appeals to the public, it will never be successful. We need to do the health economics research, so the whole price of action versus price of inaction. And then there is a whole segment of implementation research, which can be done through the center, looking at integrating smokeless tobacco cessation in primary healthcare practices, developing effective community level intervention, developing effective cessation services, and building international collaborations through multi-centric studies, which can answer these complex questions and complex problems. So as I say, the compass of research must extend from molecules to markets. And I think BCSC is in a very, very strong position to do that. We need to move, we need to do research on molecules as well as markets. And the multi-sectorality which we are talking about has to convert into multi-sector reality. And for that, transdisciplinary research is required. And just to end, if we don't create the future, the present extends itself. We should not be like ostrich, just bury our head in the sand saying that this is not a problem, while we see a storm brewing somewhere else. I think we need to be proactive, and we need to develop effective strategies. So thank you very much for listening to me, and I hope this was helpful. Happy to answer any questions you have. Thank you.
Manu, thank you very much indeed for that. That's a blast of reality, I think, for us all. Can I remind people to use the chat function to raise your questions, your Q&A function rather, to raise your questions, and we can address those at the end of the session. The next speaker in this fourth session of the afternoon is Dr. Adrian Biddle, who amongst other things has been one of the key organisers for this afternoon. I think we should, Adrian, acknowledge your input into that. For those who don't know Adrian, he's come from Bristol via the Gurdon Lab in Cambridge, where he did his PhD, to the Blizzard Institute, originally as a postdoc with Ian Mackenzie, and now as head of the Biddle Lab itself, with a major function or major research focus on cancer stem cells and heterogeneity and plasticity in cancer, particularly oral cancer. Adrian, that's the subject of your talk this afternoon, so the floor is yours.
Great, thank you, David. I'll just share my screen. Okay, so yes, thank you, everyone. This has been a fantastic afternoon so far, and I've certainly already learned a lot. My talk today is on cancer stem cell metastatic potential in models of human oral cancer. And I wanted to start by talking about how cancer stem cells fit within the 3 key research themes of Barts Centre for Squamous Cancer. So cancer stem cells have 2 important attributes. They are the subpopulation of cells within a tumour that have self-renewal potential, so can drive long-term tumour growth. And they also have multi-lineage potential, so they can take on alternative lineage identities due to their phenotypic plasticity, which can enable them to acquire attributes that drive tumour progression and spread. And because of this, cancer stem cells, let's turn on the laser pointer, cancer stem cells can mediate a progression from dysplasia to invasive cancer, a tumour spread beyond the primary site and interaction with the tumour microenvironment. And because of all this, of course, then influencing patient outcomes. I think this fits really within all of the 3 research themes of the Barts Centre for Squamous Cancer, because of course, if we want better treatments for advanced disease, we need to be able to target the cancer stem cells that drive tumour progression. For improved molecular diagnostics and prognostics, diagnostic and prognostic signatures of cancer stem cells, and particularly the different phenotypes that cancer stem cells can take on, will be really important to predict how a tumour might progress. And of course, all this must sit within the health system strengthening, because any interventions we design within the for treatment or diagnostics must be feasible within the current health system, and I think that's really important. So today I wanted to talk to you about cancer stem cells and how they mediate metastasis. And this is it's very simplified schematic here of a primary tumor with all the heterogeneity of cell types within the primary tumor. And cancer stem cells, because they have this phenotypic plasticity, can undergo epithelial to mesenchymal transition, or EMT, and invade through the extracellular matrix. It's these blue diamonds. So they undergo this EMT, which means they take on a more mesenchymal phenotype, can break away from the primary tumor and invade, they disseminate through the circulatory and lymphatic systems, and then seed secondary tumors at metastatic sites. And in order to seed the secondary tumor, they must undergo mesenchymal to epithelial transition, or MET, to seed a new epithelial tumor. So there's a sequential EMT and MET, and it's been found that the cancer stem cells which undergo this EMT take on this hybrid EMT phenotype, or hybrid EMT CSCs. In fact, they must retain the ability to switch back to an epithelial phenotype. If they undergo a full transdifferentiation to mesenchymal phenotype, they can no longer seed new tumors, and that's important. But this has been identified and researched using animal models. But the problem we have is that this process has never really been seen in human tumors. In human tumors, people see the primary tumor, but you never really see single cell invasion away from the primary tumor at the primary site. And people have looked at, have tried this. This is a study, head and neck cancer in 2017. It's a Cell paper from Bradley Bernstein's group in 2017. And they looked for markers using single cell RNA-seq, looked for markers of these hybrid EMT cancer stem cells, and then stained pathological specimens. You can see they have staining at the invasive edge of the tumor body, but still no invasion into the stranded stromal region. So we asked, I'm just going to move that because I can't see my own slide. We asked, can we study tumor spread beyond the primary site in human head and neck cancer? And this work on this slide is work performed by a PhD student in my lab, Leah Ambler, who's been very talented and done some great work. And Leah has used a model where it's a 3D in vitro tumor invasion model, where she takes oral cancer cell lines and seeds the cell into a matrix gel, which she sets the matrix gel. So matrix gel is a basement membrane extract, so it mimics the primary site of the tumor. And then she puts this matrix gel into a larger collagen gel, extracellular matrix, which mimics the extracellular matrix surrounding the tumor. And Leah then lets the cells invade out from the inner matrix gel into the surrounding collagen. And she's developed an enzymatic method to separately disaggregate the collagen gel and then the interior matrix gel, so you can separate out the non-invasive cells which stay within the primary site matrix gel from the invasive cells which invade out into the surrounding collagen. And Leah's doing all sorts of experiments on these, including single cell RNA-seq to determine a transcriptional signature for these invasive cells, which we're going to test to see if that's predictive of metastasis. But in one of the first things that, so I'm just trying to move this over. One of the first things that Leah did was to perform a flow cytometry using two cancer stem cell markers that we use in oral cancer, CD44 and EpCAM. And on the separated non-invasive and invasive cells, you can see the non-invasive cells here. This is from an oral cancer cell line. It's got a distribution that's very typical of our oral cancer cell lines, in that you have an epithelial population, this is the majority of cells, which are EpCAM positive and CD44 low, and you have a smaller population of cells which have undergone an epithelial to mesenchymal transition. These are the cancer stem cells which have undergone EMT, and these are CD44 high, EpCAM negative. Within the invasive cells, they get quite a different signature. It's quite a different profile, flow cytometry profile, where virtually all of the cells now have undergone an EMT, showing the fact that in this model, this process of undergoing an EMT is really strongly linked with this invasion out into the surrounding gel. And Leah quantified this across 3 different oral cancer cell lines, and showing that in the invasive cells, you have a much higher degree of EMT compared to the non-invasive cells in grey. And we also see this EMT population in tumours. These are oral cancer cell lines. We can look at oral tumours. We also see this is a CD44 high EpCAM low population in an oral tumour. So one thing that's was very particularly interesting, if you look here back at these flow cytometry plots, in the non-invasive cells, the EMT population you can see here is CD44 high EpCAM negative, whereas in the invasive cells, there's a much greater spread of EpCAM within this population. You've got some EpCAM positive cells, which is interesting because this goes back to this idea from the previous slide of a hybrid EMT, where cells undergo an EMT but retain some epithelial characteristics to enable them to switch back to an epithelial phenotype to seed secondary tumours. And we wondered if this is what we were seeing here. And of course, also, if these cells are retaining EpCAM, could that mean that we could use that marker to see single cell invasion in human pathological specimens? So Gihad Youssef is a postdoc in my group. Gihad has a real interest in staining pathological specimens and also in biostatistics. And so Gihad looked to see if he could identify these EMT cancer stem cells in human head and neck cancer specimens. But before going into the pathological specimens, Gihad first used this cell line model. He used a CA1 cell line, which is an oral cancer cell line, and a subline which we have derived from the cell line. We call it the EMT stem subline, and this is a subline which is greatly enriched for this hybrid EMT cancer stem cell phenotype. And Gihad stained in a multicolor immunofluorescence staining for EpCAM, which is the marker we think is the retained epithelial marker, Vimentin, which is a marker of cells which have undergone an epithelial to mesenchymal transition, and CD24, a stem cell marker, which in a previous study, I'm just showing one image from this previous study here, we showed that in this EMT stem subline, the cells which retain CD24 have the ability to undergo an MET and reseed the epithelial population, whereas the cells which have lost CD24 have lost this phenotypic plasticity, which is characteristic of stem cells, and are now restricted into a mesenchymal phenotype. So we stained with the stem cell marker CD24 alongside EpCAM and Vimentin. You can see there's a lot of heterogeneity in the parental cell line, but in the EMT stem subline, which is enriched for this hybrid EMT phenotype, you get a greatly increased amount of co-staining for these three markers. We compared this to staining with Vimentin, CD24 and pankeratin, which is a commonly used epithelial marker, which we think is more quickly lost as cells undergo EMT, and indeed the EMT stem subline didn't seem to stain for pankeratin. These cells have lost keratin expression. Gihad used, we have some fantastic automated fluorescent microscopes within our phenotypic screening facility at the Blizzard Institute, which can perform single cell analysis across multiple fields of view, and Gihad used this to quantify this cell line work and show indeed that the co-expression of EpCAM, Vimentin, and CD24 in individual cells was greatly enriched in this EMT stem subline. So Gihad then took this approach into tumor specimens. This was in collaboration with Dr. Hannah Cotton, who is a consultant pathologist at Barts Health, and Hannah pulled some tumor blocks for us. She took 24 tumor blocks initially for this initial study, which she stratified equally into non-metastatic and metastatic tumors based on the clinical characteristics. And then Gihad performed this immunofluorescence staining with EpCAM, Vimentin, and CD24 on these tumors. And you can see this is a non-metastatic tumor here. And you can see here this is a tumor body with staining for EpCAM and then the surrounding stromal region. And there's not much staining, co-staining for these three markers in this stromal region. There's very few cells. Contrast to that with a metastatic tumor here, where you've got again the EpCAM positive tumor body, and then the surrounding stroma has got in this instance here, a great number of cells which are co-staining for EpCAM, Vimentin, and CD24, suggesting that co-expression of these three markers is characteristic of metastatic tumors. And because Gihad was using these automated microscopes, he was able to effectively screen these 24 specimens and look at a huge number of fields of view and quantify the number of cells which co-stain for these three markers. And Gihad developed a technique for using the analysis software for using the EpCAM expression. If there was a large body of EpCAM that was designated as tumor body, anywhere where there was no EpCAM expression or just very diffuse EpCAM expression was tumor stroma, and she was able to separate out tumor body and stroma very nicely using this method in these fields of view that image the tumor and stromal boundary. And by doing this, Gihad quantified the co-expression of these markers, and you can see a normal region in tumor body, there's not much going on. But if you look at the surrounding stroma of non-metastatic versus metastatic tumors, there's a very stark difference in the EpCAM, Vimentin, and CD24 co-staining. There's a huge number of cells and stroma surrounding metastatic tumors co-staining for these three markers, whereas it's much less in the non-metastatic tumors. Whereas if you take away the stem cell marker CD24 to look at EpCAM and Vimentin, there's not much difference between the two now. And substituting in keratin, there's no cells in the stroma expressing keratin, again showing that keratin is lost quite quickly seemingly, whereas EpCAM is retained, and we can see this single cell migration cells away from the tumor. So this supports the idea that these EMT cancer stem cells co-expressing epithelial and mesenchymal markers alongside a stem cell marker are associated with metastatic tumors where they invade into the stroma. But as I said, Gihad has a great passion for biostatistics and particularly machine learning, and Gihad went on to develop a machine learning technique for predicting metastasis based on the staining profiles, what Gihad did, which he took the grey level at intensities for each nucleated cell in these tumour specimens and the imaging of these tumour specimens. And then the labour intensive bit was to manually create the images. Of course, there was no automated way to do this, to include only imaging tiles which had epithelial stromal boundaries, like the ones I showed you on my last slide. And he ended up with 3,500 images, imaging fields of view at 40 times imaging across these 24 tumours. He then split these into a 70% training and 30% testing set. So 70% of the images were used to train a machine learning algorithm to predict whether a specimen was metastatic or non-metastatic, and then he used the remaining 30% to test this algorithm. And Gihad tried a number of different machine learning techniques, and the most successful was artificial neural network, where you input the grey level intensities of momentin, CD24, and EpCAM. He also tried pancreatin as a control, and predictably pancreatin didn't work, but with momentin, CD24, and EpCAM as inputs, this then trains these hidden layers to output a prediction of whether this is a metastatic or non-metastatic tumour. And in the testing set, Gihad got an area under the curve of 87%, which is really good and actually better than published efforts. So that's really encouraging in this small set. And Gihad is this small initial set is now published in bioRxiv for April 2020. And Gihad is expanding the study with a greater number of tumours that Hannah has found for him, and to really increase the robustness of this machine learning approach, which we hope could be used as a predictive method alongside some of the other methods we're developing, which we'll mention towards the end of the talk. So that's really exciting, and we are continuing that study. And I wanted to switch now to talking about a different study. And this is Alice Skamama, who is a PhD student in my group, who's working on this really exciting project in collaboration with Julian Gautreaux at the School of Engineering and Material Sciences at QMU, our Mile End site. And Alice is using a microfluidic chip. So this has 3 microfluidic channels, a central channel where she puts in a 3D matrix, and then she can put different cell types on the 2 side channels. And she's using this to look at how cancer stem cells mediate the interaction of the tumour with the microenvironment. In particular, in Alice's case, she's putting HUVEX into the left-hand channel and letting these vascularize the 3D matrix, and then putting head and neck cancer cell lines in the far channel, and watching how these interact with the HUVEX and how cancer stem cells mediate this interaction. And this is an image here from some Alice's work, where she's, this is in red here, this is the vascular network. And then in green, you've got GFP marked cancer cells, which you can see how they're interacting with the network as they invade into the matrix and then intravasate into the vascular network. And therefore, we can look at the role of cancer stem cells in mediating invasion and interaction with the vasculature in intravasation. And also, interestingly, if this is a picture here, a bright field image of one of Alice's chips, where she's got 3 channels, and she's got this matrix in the central channel here between these posts, and she's got a vasculature, and you can see the vasculature here. And if she lets the vasculature develop to the point where it goes all the way across, she can put in the cancer cells on the right-hand side, and watch how you can see them here flowing into the lumen of the vascular network. And that means you can actually look at the opposite, the other process of extravasation at a secondary site, and investigate how cancer stem cells might aid that process of extravasation. So we're really excited about this project, and because it will help us understand more about how the tumour cells interact with the vasculature, the tumour microenvironment, and how that's maybe mediated by these cancer stem cells that can undergo these phenotypic transitions we've been talking about. We also have some other future projects which are coming up soon and starting soon. We'd like to translate a predicted signature towards a clinical setting. So we've talked about Gihad's immunofluorescence staining. I talked about feasibility within a clinical setting. It may find that multicolor immunofluorescence isn't the best or most feasible method to use in a clinical setting. So we're also looking at other methods. Leah and Gihad together, we talked about Leah Amda's work, where she's been making these 3D gels where she can isolate the invasive cells, and Gihad has been working on machine learning algorithms. And so Leah has developed a transcriptional signature for the invasive cells in her model, and her and Gihad together have been working on a machine learning approach to optimize a signature that's predictive of a transcriptional signature that's predictive of metastatic status in oral cancer. This could be be really feasible, actually, clinically, potentially, and they're now testing this. Gihad and Leah are now testing this signature on these this library of metastatic and non-metastatic FFPE specimens that we have. And if that works out well, then we'll look to translate that towards a clinical setting. So I think a transcriptional signature of cancer stem cells in oral cancer could be very valuable to help predict whether the likely course of tumor progression and design therapies according to that. We have a new project starting in the autumn, a new MRC project, MRC funded project, that is going to be looking at therapeutic inhibition of metastatic transitions. Here we're going to use a single cell RNA-seq based methods to really try to understand the transcriptional networks, the molecular networks underlying those EMT and MET transitions, which is so important, we think, for metastasis, and then look to therapeutically inhibit those transitions and then see how that plays out in our in vitro metastasis models. We have a clinical fellow joining us in the autumn who will be incorporating immune cells into the microfluidic metastasis model. At the moment, we're just using a vasculature, and we're really keen to increase the complexity and incorporate immune cells into that. And particularly to look at the genomic signatures of different cancer cell line panels and see how the genomic status of the cell lines can influence interaction with these immune cells and how these immune cells interact with the tumor. And particularly having just seen Jennifer Grantis' talk, it's particularly exciting think about how different genomic signatures of the tumor may interact with the immune system there within this in vitro microfluidic model. And also, of course, how cancer stem cells might mediate that. And finally, we have a rotation student in the lab at the moment, Sophia Lunetto, who has been working on building biophysical models based on, she's been using the microfluidic metastasis model to understand how the different patterns of invasion, invasive cell morphology within this model, and using that to build a biophysical model of tumor invasion, which we then will try to use to predict tumor behavior in human specimens. So again, take data from our in vitro models, use a data-driven approach to model that, and then apply that to tumor specimens and see how that goes. So that's, I think, very exciting as well. Sophia has been joining us for a rotation to do that. So I just wanted to finish. I started with a Venn diagram, so I thought I'd finish with one. I talked initially about how I think cancer stem cells can fit very nicely within the 3 research themes of the Barts Centre for Squamous Cancer. And now to finish, I want to talk about how I think Barts Centre for Squamous Cancer will really be really exciting and important initiative to support my own lab's work and bring together some key research streams of QMUL. So I think hopefully you've seen, as I've talked about the work we're doing, that we really depend on using in vitro models that are predictive of what's happening in human tumors, on using human tissue and clinical data to validate this, and on data science for our we're using our machine learning based approaches for clinical prediction, biophysical modeling, and also, of course, we need data-driven approaches to handle the vast amounts of transcriptomic data we're producing. And these 3 areas, predictive in vitro models, human tissue, and clinical data, and data science, are really key, really big research trends at QMUL. And we're really lucky to be working at QMUL, I think, QMUL, I think, because the university has these great research strengths. And this is underpinned by, I've just noted around the sides here in this smaller writing, underpinned by these research centres and resources which underpin these 3 research strengths of QMUL. And I think Barts Centre for Squamous Cancer will be really crucial and exciting because it will bring together these and really harmonise these research strengths into an approach that can help us target squamous cancer, because it's going to have resources to support collaboration between lab scientists and data scientists. It's going to coordinate squamous cancer tissue and clinical data, which will be really important to have that cancer tissue and clinical data, and coordinate squamous cancer experimental models. And I think this is going to be great and really exciting for our own work. So I'm very excited about the start of this new centre. And I'd just like to finish by acknowledging those who've been involved in this work. In the Blizzard Institute, I talked about Gihad Yusuf's work and Leah Ambler's work and Alice Kamama's work. Luke Gammon runs the Blizzard phenotypic screening facility, which houses these automated immunofluorescent microscopes, which we use for a huge amount of our work, as you've probably noticed. And Luke's incredibly helpful, incredibly knowledgeable, and really helped with all of that work. Ian McKenzie and Mike Philpott are 2 colleagues who've helped enormously to help me get my lab off the ground and going. I'm very appreciative of them. And I talked about Sophia Lunetto, who is a new rotation student in the lab who is doing the biophysical modeling. We have some great collaborators, Hannah Cottam and Kim Piper at Barts Health, who've been collaborating with the tumour specimens, and Julian Galtro at SEMS, who has been generating the microfluidic model and helping Alice bring that technology over to the Blizzard Institute. And the new projects I mentioned, MyTech Tear in the Institute of Dentistry has great experience with developing molecular diagnostics, and he's collaborating on that. and Edward Haneza at ICT Austria is helping us with the biophysical modelling. And of course, I'd like to thank our funders, Animal Free Research UK, Oracle Cancer Trust, and the National Centre for the 3 Rs, without whose generous support, none of this would be possible. So thank you, and yeah, I'll stop sharing my screen now. Thanks.
Adrian, thanks very much indeed. We now have a few minutes for questions before I ask David Kelsall just to bring the afternoon to a close. I've got a question for Manu, first of all. Manu, you raised a really troubling issue for me, I think, which is, I mean, in part, it's a low middle income country problem in that the disease exists at a high prevalence in areas where the research capability is disproportionately low. How do you see that being impacted? And do you see that somewhere like Barts Centre for Squamous Cancers has a role to play in that? I mean, perhaps related to the prevalence of the South Asian community in East London, for example. And you're muted at the moment.
Sorry, Professor, it's the perils of working online. Well, thank you for a great question. And I think Barts Centre for Squamous Carcinoma, I think, is at a very, very strategic position. And I think this funding comes at a very strategic moment. As I said, that there is a need to establish these kind of transdisciplinary research studies. And there cannot be a good place to do this, but in the area where QMUL or the Blizzard Institute and the Institute of Dentistry is located. Now, this being said, there are various opportunities, and I would like to just spend a few minutes on that. The opportunities lies in developing population level studies, and again, as I said, looking at epigenetic features. What are the genetic mutations which happen because of the harmful habits of smokeless tobacco consumption, smoking, alcohol, and how these habits cluster together and bring about changes which again then impact on the survival rates of oral cancer, on the disease progression or the disease severity. We heard Professor Coulthard's presentation that survival rate is very less, and of course the treatment is very extensive. So there are various factors. So starting from a molecular level, going up to a public health level, to an implementation research level, there are tremendous opportunities to establish or nest some very good population-based studies, and you have the right population base also to do this kind of study. As I said, this is a problem which is of a South Asian origin, and it is increasingly prevalent in South Asian populations. But this is not a problem of South Asia. This is increasingly becoming prevalent. You can see people in United Kingdom, roaming on the streets with pouches or packs of tobacco with them. So it's actually something which has to be looked into, and something which has to be studied, something which has to be seen from an inequalities perspective, something from a health promotion perspective, something from the molecular side of things also. So it's an agglomeration of different disciplines to bring about a robust policy level change, I think, and BCSC is at the right position to do that.
So Adrian, I wonder if I can link that to a question for you. You talked about the translational impact of your research and how you can use that to improve diagnosis and move towards that sort of area of precision medicine. How scalable do you think that is, and over what kind of a time scale to begin to make an impact on improved diagnosis and more targeted treatment?
Yeah, that's a really good question. And I think the key thing probably for us is to make sure at this stage we're communicating with clinicians and also with patients, of course, about the feasibility of the approaches. So whatever we're designing now is something which is going to be feasible within the system. So I think that's where a transcriptional signature, we've seen that in breast cancer with things like EndoPredict, a transcriptional signature has been really valuable for predicting prognosis and designing treatment on top of that. And I think that's something where we're looking at designing a transcriptional signature, which we can then test initially on a cohort of tumor specimen, of a library of archival specimens, and then take that into into clinical trials. Of course, there's good funding schemes for that. So I'd hoped that within within a decade we may have something where we can offer that's been tested clinically and which can help predict the potential, the course of head and neck cancer, and therefore help design therapy. So of course there's the diagnosis based in, for example, in primary care, maybe in a dental surgery, is a slightly different matter, but it can then also maybe be useful there, where maybe from brush biopsies or small biopsies we could do a transcriptional profile. And I should mention Tech's work at MyTech, who works within the Institute of Dentistry at Clonal, who's been looking at a diagnostic signature using transcriptional profile, and his work's actually quite a bit more advanced, and he's already tested that in several cohorts, and that's looking really encouraging.
Thank you very much. And at that point, David, I'll hand back to you perhaps with a question as well, which comes from Roshan Pradeep from the University of Peradeniya, who says, what are the ways to collaborate with Barts Centre for Research in the field of oral cancer? Thank you.
Well,
there's an invitation.
Yeah, I think email us and we will collaborate. I think this symposium has shown that we've had delegates from all around the world, being part of this and asking questions. So I think engage, and science is a global thing, and we can work together across the waters and do stuff together. So yeah, please feel free to get involved. So that brings the end to our symposium, our first symposium for the Barts Centre for Squamous Cancer. I think it was a very informative and enjoyable experience with a real diverse array of talks, but with some overlapping themes. And I really would like to thank Jennifer Grandis for her superb keynote lecture, absolutely stunning, and you can see real sort of therapeutic avenues that could actually be used to improve the management of SCCs. I'd also like to thank our two external speakers, Manu and Bartimo, again, superb talks, and of course our own, Inesh and Adrian, for their excellent talks as well. And I think this afternoon really does show the breadth of research that's happening across the SCCs. Again, I'd like to thank all the participants who've been involved and stayed to the end. I think we had about 140 or so people during the day. So in conclusion, I'd like to thank, on behalf of the executive, Irene, Paul and Catherine and Adrian, to say thank you for helping to make this symposium a superb start to the launch of our new Barts Centre for Squamous Cancer. And we look forward to seeing you all at our next symposium, which hopefully could actually be happening at the Blizzard Institute. So on that note, I thank you all and have a good evening.